so okay, here we go.
Cool. Hey everyone, how's it going? All good? Awesome. We're going to be doing the exam for review. I'm taking lectures 13 and 14. Alex is 15 and 17, and then Shrey will make a guest appearance for lecture 16. How do I like?
You have to click the slide, sorry. It didn't loom as stupid like that.
No worries, no worries. Okay, some exam logistics before we get started. So the exam, you should probably know this by now, but it's this Wednesday, 6 to 7:20 p.m. There's locations. These are also listed on Canvas. We're also having a Jeopardy and problem-solving session. I know this is mainly like content review stuff. So if you have specific questions, if you want some experience with solving problems, that's going to be on Tuesday, 5 to 7, and that's in this room. We also have office hours happening right now, I think in 163? 66, okay. Personally, 166. So if you have any questions and you want to head over there, feel free. Y'all got this. You've done 2 of these. You can do 1 more. We're here to help you. Ask questions of us. Ask questions on Ed. Come to office hours. Yeah?
I will say that once you get through this exam 4 is going to be much more chill.
Yes.
In terms of content, the sheer amount of content is going to decrease a lot for exam 4. So you just got to get through this one.
Yep. So close. No more pathways. Very few pathways after this. So the end is in sight. Lecture 13. This is all the lipid stuff. Here's a bit of some objectives, a bit of overview. We're going to be talking about the types of lipids and other structures. So we're talking about, well, that whole list there, also sterols, steroid hormones. We're also going to talk about membranes because those are made up of lipids. We're going to talk about membrane behavior, the structure of membranes, the fluid mosaic model, which is something you probably heard about in something like Bio 200, but we're going to go a little bit more in depth in that. Other than that, we're also going to talk about getting stuff across the membrane, both in terms of moving molecules around and also transporting signals across and through membranes. So the basic lipid structure are these things we call fatty acids. The fatty part indicates this large hydrophobic tail, and the acid part comes from the carboxylic acid head. That's polar, that's hydrophilic. So you have a polar head, hydrophilic head, and a hydrophobic tail. That ends up being important for membranes, the structure of membranes. We'll get into all that later, but good to know. Saturated fatty acids mean that every carbon has as many hydrogens as possible. There's no double bonds in these structures. And unsaturated fatty acids have at least 1 double bond. These unsaturated fatty acids tend to be in the cis configuration. Also, having unsaturated fatty acids in the membrane is useful because it increases the fluidity. Again, we'll talk about that later, but yeah. So when we're talking about the structure of these fatty acids, you don't need to worry about naming the specific fatty acids, but it's nice to have a way to indicate the structure of them. And the way we do that is with this little ratio type thing. So it's a ratio of the number of carbon atoms to the number of double bonds. For example, a 12-to-0 fatty acid has 12 carbons and has 0 double bonds. If the fatty acid does have a double bond, we indicate the position of that double bond with a little delta sign, followed by the number of that position, or the position of that bond. So if you have an 18-to-2 delta 9-12 fatty acid, that means you have carbon-carbon double bonds between carbons 9 and 10, and between carbons 12 and 13. When we're counting, we are starting from the carboxylic acid carbon and going down the chain. The 2 most important ones that Professor Solomon specifically mentioned in lecture are these 16-carbon palmitic acid and the 18-carbon stearic acid. 16-carbon palmitic acid is the most important one. We will talk about this again later in fatty acid biosynthesis. All right, not much to say here. This is just sort of a useful graphic for visualizing the lipids, the structures that we are going to talk about. These molecules are going to have different numbers of fatty acids. They're going to have different backbones, and they're going to have different head groups, sort of substituent type things. We'll get into more detail about all of that. Yeah, so for identifying these, some things to be comfortable with are recognizing lipid structures, categorizing them, following that chart that we had there. Also, pointing out specific functional groups that we learned about in lecture is something that you can definitely be asked on the test. And also identifying the functions of these lipids. Again, we'll get into detail about this, but something like triacylglycerol is used in storage. Other lipids are used in membranes. Useful stuff to know. All right, the first structure we're going to talk about is waxes. So waxes are ester linkages between a fatty acid and a fatty alcohol. These images here are examples of those. The left structure is a fatty acid. The right structure is a fatty alcohol. These are used by plants and animals to form waterproof barriers because of the hydrophobic characteristics of these waxes. An identification tip for these is you're just looking for two hydrocarbon chains joined together by an ester linkage. That's, again, fatty acid, fatty alcohol. Those are joined together by an ester linkage. Also, for all of these slides that mention specific kinds of structures, that text in blue is just like an easy way to identify them. There's a summary slide for this as well, where we will highlight sort of specific things that you can use to tell these apart. But yeah, just a thing to keep an eye out for. So the next structure is triacylglycerols, or tags. These lipids are used for storage. They're also called neutral fats, and they're also called triglycerides. The structure of these is 3 fatty acids, and each of these fatty acids is joined by an ester linkage to glycerol. Glycerol is a super important structure to know for this exam. It is the backbone of many things. We're also going to talk about breaking it down, getting some energy from it later. That's what it looks like. You've got this sort of 3-carbon skeleton and 3 hydroxyl groups attached. So when you're making a triacylglycerol, again, 3 fatty acids, each joined to the glycerol molecule. The next structure that we're going to talk about is glycerophospholipids. These are seen primarily in membranes. Glycerophospholipids are made up of a glycerol, again, that same structure as on the previous slide, as well as 2 fatty acid chains. These are the sort of hydrophobic tails of a membrane lipid. If you see sort of like cartoons of membrane lipids, it's got like the circle head and those 2 little stringy things. That's what these are, fatty acids. These are going to be hydrophobic, and the head groups are going to be hydrophilic. That's important for structuring membranes. That's how membranes form, things like that. Yeah, the head group, again, hydrophobic. He mentioned most specifically phosphatidic acid. That's the glycerophospholipid with a hydrogen as its head group. You do not need to memorize all the different kinds of glycerophospholipid heads. Just know that there are different kinds. Know that the naming convention, things like that. It's a huge list. It's not that important. When you're identifying these, you're going to look for these 2 fatty acid chains. These are joined, same thing as before, with an ester linkage to the glycerol. And you have a phosphate attached to a head group. Remember, that head group can be a bunch of things. So those glycerophospholipids are what we use to make membranes. Membranes are made of fatty acids. We use this to separate the inside of the cell from the outside of the cell. And we also use this to separate the inside of the cell from different parts of the inside of the cell. Intracellular compartmentation. We said it before, important to drill this, lipids have a hydrophilic head group and a hydrophobic tail, and that is going to drive what forms these lipids. You can also embed proteins within these membranes based on their characteristics. The way that these membranes form is driven by the way that Professor Solomon describes it as maximizing the entropy of water. So if we have fatty acids by themselves, sort of on this left diagram, that state is thermodynamically unfavorable because there's a lower entropy state of water in that configuration. By contrast, if you pack these together really tightly, that becomes a lot more thermodynamically favorable for water. So an important detail to know about this is that the driving force of forming these membranes is the entropy of water. Another format of these, when you have a high enough density of lipids, are these things called micelles. Micelles, this is sort of like a cut-in-half way of visualizing it, but micelles are like a sphere, all made up of lipids. You have the hydrophilic head group pointing out, the hydrophobic tails pointing inside. Again, that is maximizing the entropy of water. That's like the sort of best-case scenario structure for the entropy of water. All right. Lipids with 2 fatty acid chains. Again, we talked about these glycerophospholipids are what make membranes lipid bilayer. So we have these sort of 2 rows of membranes, one pointing out, one pointing in. These individual units, again, held together by the entropy of water. You have non-covalent interactions within the membrane, so with those lipid tails, and hydrophilic interactions between the head group and the water, the sort of exterior of the cell. again, you've seen this before in Bio 200, but it's nice to know sort of what's forming it, what makes it up, things like that. I don't know what Trey's doing with this slide. But yeah, this is a slide that Professor Solomon put up here, but you don't need to know the details of the slide, but these are just different ways that cells apply compartmentalization. We learned some of these in the last unit. This also includes things like micelles, membranes that we talked about in the previous slide. Okay, the next type of structure is glyceroglycolipids. As you can tell by the name, these are pretty similar to glycerophospholipids in structure. The only difference is that instead of having a phosphate attached to a head group, our sort of head group is just a sugar structure. So these, again, made up of glycerol as the backbone and 2 fatty acid chains. Your head group is a sugar residue. And the easiest way to identify these is if you see 2 fatty acid chains and you see a sugar head group, glyceroglycolipid. Another structure, these are seen in archaea, are ether lipids. These are made up of 2 fatty acids, but the difference here is instead of an ester linkage to your backbone glycerol, you have an ether linkage. These also have a head group substituent similar to glycerophospholipids that can be changed into a bunch of different things. Easiest way to identify these, look for the ether linkage instead of the ester linkage. Yeah, the bottom diagram is a bit confusing. I was definitely confused by this when I first saw it. But it's just an example of 2 of these lipids associating with each other, creating their own type of sort of built-in membrane. The structure is extremely favorable. It's extremely rigid and stable. So archaea use this to withstand extreme temperature conditions, extremely high and extremely low temperature conditions.
And to also add on top of that, that structure on the bottom is actually the same motif as wax. So there's 2 ether linkages on the sides. So in the bullet point right there, it's extremely rigid and stable in terms of extreme conditions. That's why we use a lot of like wax, like chapstick and whatnot, so that way our lips don't become chapped.
Yeah, good way to memorize it. One of our last structures, we're getting through them. Sphingolipids. The backbone of these is sphingosine or sphingosine. I don't actually know how to say that. These are found mostly in neurons. The easiest way to identify this is you're looking for a backbone that has nitrogen. Glycerol does not have any nitrogen. If you see a backbone with nitrogen, it is more than likely a sphingolipid. These also will have 1 fatty acid. That's another way to identify them, as well as a head group substituent like the other lipids. Yeah, again, little identification tips in blue, useful for studying. These slides are uploaded on Canvas in case you need them. All right, phosphatidylinositol. This is a molecule with a name that is way too long that also confused me for a very long time when I took this class. We'll go over basics of this, but phosphatidylinositol is used for cellular communication. The way that we make these is by using phospholipase enzymes to cleave the bond between the oxygen of the ester and our substituent groups. The one specifically mentioned by Professor Solomon in lecture is this phospholipase C. That's this here. It's going to cleave this bond. And what that is doing is we are forming diacylglycerol. So that's this structure on the top here. You have glycerol with 2 fatty acyl chains, hence diacylglycerol, and inositol trisphosphate, IP3. The structure that we are starting with here is phosphatidylinositol bisphosphate, also known as PIP2. That's the abbreviated form. So on the next slide, we'll go into like a diagram that mentions this, but the sort of overview is that we're going to use IP3 and DAG to activate protein kinase C, which will in turn lead to enzyme regulation. Here we are, a bit of a crazy diagram, but we're going to walk through all the steps. So the first thing that's happening is that you're having a hormone binding to the specific receptor. That's an extracellular reaction. As a result, you're going to have an activation of this protein here. That protein is going to go off and bind to phospholipase C, which is that enzyme that we talked about on the previous slide that is cleaving this bond right here, forming IP3 and DAG of PIP2. The next thing that's going to happen is that you have your IP3. IP3 is going to run over into the endoplasmic reticulum, and it's going to open calcium channels. What those calcium channels will do, of course, is release calcium. Calcium will travel over along with DAG, and both of those will in turn activate protein kinase C. Protein kinase C is then going to phosphorylate other enzymes, do its own thing. We don't need to know the details of that. The biggest idea of this slide is, of course, knowing the specifics of IP3 and DAG, what they're doing, where they're going, and also, we'll talk about this a bit more later, but hormonal signaling. So a hormone is binding to the outside of the cell, causing conformational changes and conveying a signal. All right, sterols, our second to last structure, I believe. These are going to have a pretty complicated ring structure as well as a polar head group. That's this OH right here. Sterols, especially, the one that we're caring most about for this test is cholesterol. Cholesterol has a ton of branching. You just have a bunch of hydrocarbons here. The easiest way to identify this, the only one that looks really similar, is your steroid hormones. So in terms of differentiating from lipids, if you look for those 4 rings, that's a pretty clear indication that you have either a sterol or a steroid hormone. We're going to talk about this later. I keep saying that, but you will have to know how cholesterol is going to impact membrane fluidity, and also how cell signaling differs when we are using steroids and steroid hormones. Sterols and steroid hormones, excuse me. Here's the membrane fluidity thing. The way that Professor Salman put this is that it increases the range of temperatures at which your membrane is stable. So it sort of acts as a membrane fluidity buffer. You want sort of less extreme changes, and that's what cholesterol is going to do. So what happens at high temperatures is that your lipid chain is going to vibrate a bunch, move around. That's going to cause the membrane to fall apart. That's sort of an overfluidity, I guess is the way to think about that. And the way that cholesterol will help mitigate that is by fitting into the gaps between the lipids, and it will add stabilizing interactions between them. That's going to raise the melting point of the membrane and stabilize the fluidity. Cholesterol can fit both in between these lipids here. Oops. And also, it's not shown in this diagram, but it can fit in the gaps of unsaturated lipids. See how that structure is sort of like branching out? Cholesterol can fit into there as well. So that's what we do at high temperatures. What we're going to do at low temperatures is, well, first, what's happening is that the membrane is vibrating less. It's becoming brittle. You have sort of a decrease in fluidity. The solution to that is that cholesterol is going to continue to associate with these fatty acids and is going to increase the fluidity of the membrane. So at high temperatures, you're decreasing the fluidity so it doesn't fall apart. At low temperatures, you're increasing the fluidity to maintain that movement and, yeah, that characteristic. Okay, our last structure, steroid hormones. Steroid hormones are all derived from cholesterol. That's the most important detail of these. You do not need to know these specific structures. It is important to know that they come from cholesterol, though, which you can also sort of tell from the 4 rings that we have. The other thing that is different between steroid hormones and sterols is that we have polar groups on both sides of the molecule. So in cholesterol, we had an OH right here. In these steroid hormones, we're going to have OHs on both sides, or just in general, these polar groups on both sides of the molecule. That's going to allow them to walk straight through membranes and deliver signals. It's because they have both polar and nonpolar character, just like those membranes that we just talked about. Again, you do not need to know the specific names of these, just be able to recognize that they are steroid hormones and also differentiate them from cholesterol. The easiest way to do that is that this has 2 polar groups or more, whereas cholesterol just has 1. Membrane fluidity. I think these are the best names of any enzyme ever: flipase, flopase, and scramblase. So another key feature of that fluid mosaic model is the fluid part. So these lipids can move within the membrane laterally, side to side, and that can happen very quickly. But moving from the outside to the inside or the inside to the outside happens very, very slowly without the help of an enzyme. So we're going to use these specific enzymes to flip-flop them. Flipase is when we're going to take a lipid from the outer leaflet and we're going to move it into the inner leaflet. And flopase is moving a lipid from the inner leaflet to the outer leaflet. Both of those are driven by ATP hydrolysis. They take energy. The last one, which does not require ATP hydrolysis, is scramblase, which is where you take a lipid from the outside and a lipid from the inside, and you're going to swap them. Yeah. Yeah?
Do you have an immediate example of why you want to flip around your lipids in your membrane?
That's a good question. I am not sure. One thing that I can think of is possibly that if you have lipids that have, for example, different lengths of chains, or one that's saturated, one that's unsaturated, and for whatever reason you want a specific characteristic on the outside, that you want to flip those.
The best example I can give is you're trying to make vesicles. We know that they're circular, but they do maintain that bilayer. So in this case, you see that they're straight. But if you, let's say, for example, you favor the flip-based activity, it's going to flip them inward and almost cause the curvature to go around and ball around. So that's an instance where you would only want flip-based and not flop-based or scram-based activity to occur.
Cool. All right, lipid composition. So the other way that we can impact membrane fluidity, other than the amount of cholesterol that we have in the membrane, is by changing the lipids that we have. If we have longer chains, longer carbon chains in these lipids, we're going to have more hydrophobic interactions. A simpler way of thinking about that is that they're just going to cluster more tightly together. That force that's acting upon them, again, that's water. The polar interactions with water is going to sort of push them more tightly together, and that's going to make that membrane less fluid. So as you increase the length of the carbon chain, your melting point is going to increase. Again, you're more tightly packed, so you need more energy to disrupt that solid state. On the other hand, if you increase the number of double bonds, the melting point is going to decrease. The membrane is going to become more fluid. That's because those double bonds create sort of kinks. It's going to spread everything apart. The lipids are then less tightly packed together, and as a result, there are less hydrophobic interactions. That ability to sort of change the fluidity of the membrane is going to allow for physiological regulation of membrane fluidity. If you increase in temperature, we're going to adapt by putting in longer lipids. Cholesterol is going to do its thing to sort of mitigate that change. You want to increase the melting point, and you want to lower fluidity so that your membrane does not fall apart. All right, here's that summary slide I was talking about. This is pretty useful for studying, I think. It's just the things highlighted in blue are the traits that are unique to those specific structures. Take a look if it's useful. We won't go through it again, but yeah. All right, transporting molecules across the membrane. There's a bunch of different things that do this. The sort of generic transporter is things that allow ions and nutrients to go through. We have receptor enzymes, and those are going to convey chemical signals when something binds to the outside of the membrane and causes a conformational change on the inside of the membrane. We just saw that with the IP3 and DAG pathway, where a hormone was binding to the receptor on the outside, causing a conformational change on the inside of the cell. The last one is a gated ion channel. These are going to allow ions to pass through, but they're more easy to regulate. They're regulated by the binding and unbinding of some kind of ligand on the outside of the cell. There's two kinds of proteins that you find, or two categories of proteins that we find associated with membranes. The first one is integral membrane proteins. These are just things that are firmly associated with the membrane. You're going to need a more harsh treatment to disrupt the interactions holding it together. These are often embedded in the membrane itself, but they are not always embedded in the membrane. I know Professor Solomon mentioned these ones that are sort of attached with like a chain. These are those ones that are anchored by covalently attached lipids. Those are also considered integral membrane proteins. So these are defined by the strength of their interaction with the membrane and not necessarily the way that they're existing. The easiest way to get these out, this is something that can definitely be tested, is like what can you use to disrupt the interactions and free these proteins from these membranes. For integral membrane proteins, you need stronger things. That includes organic solvents and also high temperatures. If you have ones that are not embedded in the membrane itself, you need something like a lipase or a deacylation enzyme. Peripheral membrane proteins are the other kind. These are the membrane proteins that are associated with the membrane but are not necessarily associated with as strong of an interaction. That includes hydrogen bonds, electrostatic interactions. These can also be associated with another membrane protein, so they don't have to be immediately attached to the membrane. Just an interesting note. These are much easier to sort of separate. You only have to break non-covalent interactions. You can change the pH. You can use a chelating, thank you, agent, or you can carbonate. You don't need to know the specifics of what a chelating agent is or what chelation is, but it's just sort of interactions between ions and metal ions. So you can recognize the amount of times that a protein crosses through the membrane. You can recognize proteins that do cross the bilayer because of these stretches, these long stretches of hydrophobic amino acids. On the inside of the membrane, again, remember those tails are hydrophobic, so things inside the membrane are going to have hydrophobic character, hence the hydrophobic amino acids that are in the membranes themselves. These hydropathy plots are definitely something that we could see a test question on. You're plotting the amino acid position, that's on the x-axis, versus the hydrophobicity of the amino acids. So any sort of peaks above the 0 line indicate a transmembrane segment of amino acids. You could see a question that's like, how many times does this protein cross the membrane? And in this case, it would be 7. You have 7 of these little peaks. This one's a bit confusing because they're sort of 2 merged together. On a test, you wouldn't see something like that. These would be pretty distinct peaks. All right, this is the sort of summary of that. This is the fluid mosaic membrane model. This is just an idea that proteins can move around in that membrane. They can redistribute as necessary. Membranes are flexible, they're adaptable, they're dynamic, both with the lipids themselves and the components embedded in the membranes. Like mentioned on that previous slide describing integral and peripheral proteins, you should be able to specify what you can use to remove a specific protein from the membrane. There's a couple kinds of transport. Again, you've probably seen something like this in Bio 200. Uniport is you're moving one molecule at a time, and that's moving down the concentration gradient. Symport is you are moving 2 molecules. One of them is going down their gradient, one is going up it. And antiport is, again, you're moving 2 molecules, except this time they are in opposite directions. Again, one is going down its concentration gradient, and one is going up it. Symport and antiport are examples of cotransport. That's just you are using 2 molecules. You're using one that's going down its concentration gradient to move another one up its concentration gradient. All right, there's a bunch of different kinds of transport as well, in addition to the kinds of transporters. There's simple diffusion, which is just a molecule going down its concentration gradient, and it's just sort of walking through the membrane. It's small enough, it's not charged, so it can just sort of diffuse through. You also have facilitated diffusion, which is molecules that can't necessarily diffuse through the membranes, so you're creating these sort of like channels. These are also going down their concentration gradient, but they are going specifically through a transporter. And then active transport is you are taking something up a concentration gradient. Primary active transport is driven by ATP hydrolysis. You're hydrolyzing ATP to get enough energy to sort of push that thing back against its concentration gradient. Secondary active transport is what we talked about on the other slide, but you're moving another ion down its gradient to bring something up the gradient. The last thing on this diagram is ionophore mediated ion transport. This is just taking an ion, surrounding it with something that can go through the membrane because the ion itself can't, and then that thing is going to carry it through the membrane and release it. It's also useful to know the examples of these that he mentioned in lecture. I know last year there was a test question that was like, glucose transport is an example of blank. So yeah, useful to know these examples that he mentioned. The first one, this is your example of facilitated diffusion. This is also uniport, you're moving 1 molecule. This is glucose transport into erythrocytes. All that's happening is your glucose is binding from the outside of the cell. That binding is going to trigger a conformational change in your membrane protein. It's going to sort of flip, and it's going to point towards the inside. Glucose is then going to unbind, diffuse into the or move into the cell. That unbinding event is going to cause it to return to its configuration where it's open towards the outside, so it can do its thing again. And we'll see that motif with other kinds of enzymes, like in this example. This is the example he gave of primary active transport. This is sodium-potassium ATPase. What's happening here is that on the outside, sodium ions are binding to the transporter. You are going to, this is again, primary active, so you are hydrolyzing ATP. The hydrolysis of ATP is going to cause a conformational change in the enzyme. It's going to go from facing the inside to facing the outside. And then those sodium ions are going to go on their way. You then have potassium that will bind. So right now the enzyme is phosphorylated. The hydrolysis of ATP caused the enzyme to be phosphorylated when it's open towards the outside. Potassium is going to come in and bind. Conformational change, the enzyme is going to go from facing the outside to open on the inside. That is triggered by a dephosphorylation, and then potassium goes on its way, and the same thing can happen again. The last example that he mentioned in class is secondary active transport. This is glucose transport into intestinal epithelial cells. So what's happening here is that glucose is entering the cell via symport with sodium ions. The sodium ions are moving down their gradient, which we need because glucose is moving against its gradient. Then you have glucose in the cell. You can do what you want with it. You're going to take sodium and you're going to take it out with antiport. So you're going to take sodium out because you're going against its concentration gradient. There's more sodium outside the cell than inside the cell. And you're going to bring potassium in at the same time to be able to move those sodium ions in. All right, that's all of lecture 13. Any questions on that? Cool. If anything comes up, again, we have office hours in, I forgot the room again, 166? 166. You can always post on Ed. We'll move on to lecture 14 now. All right, here's the overview slide of lecture 14. We're going to talk about lipid metabolism. We're going to talk about taking glycerol from lipids and turning that into usable energy. We're also going to talk about taking the fatty acids and turning that into energy. That's going to happen with beta-oxidation. There's some problems that we can find with fatty acid breakdown. That's where you have double bonds in your fatty acid chain, an odd number of fatty acids, things like that. So we'll talk through that. We're also going to talk about ketogenesis, breaking down ketone bodies, all this other stuff. All right, so the form of lipid that we use for storage, again, from the last lecture, is triacylglycerols, or TAGs. This is a glycerol, and it is bound to 3 fatty acid chains. So the first thing that we're going to do, it's not on this slide, but you're going to take a triacylglycerol, and you're going to break it down into your fatty acid chains and the glycerol. That's done with the lipase enzyme. So we want to maximize energy as much as possible. We're going to take the glycerol and use it for something. We're going to take the fatty acids and use them for something. So how we get glycerol energy out of glycerol is we're going to want to throw it into glycolysis. A theme throughout this lecture and sort of throughout nature as well is that if we can take this molecule and make it into something else that the body recognizes, we don't have to have a whole separate pathway to break it down, things like that. So what we're going to do is we're going to take that glycerol and we are going to make it an intermediate of glycolysis, glyceraldehyde 3-phosphate. The first step of that is phosphorylating it. That gets you L-glycerol 3-phosphate. Glycerol bound to fatty acids is in the L configuration, but glycolytic intermediates are in the D configuration, so we have to change the stereochemistry. The way that we do that is we oxidize, and then what Professor Solomon calls stereoselectively reduce. I don't know what that means. I don't think you need to know what that means. All that we're doing is we are oxidizing to remove the stereochemistry from the molecule, and we're reducing to reintroduce stereochemistry in the way that we want, which in this case is the D configuration. So we do that, we oxidize, we stereoselectively reduce, and we end up with glyceraldehyde 3-phosphate in the D configuration. That's a glycolytic intermediate. We can throw it into that pathway and get some energy out of that. Next is what we're going to do with the fatty acids. This is a lot more complicated, unfortunately. There's a bunch of steps of this. The first thing that we're going to do to prepare fatty acids for breakdown is we're going to activate them. The way that we do that is with AMPylation. We're just adding an AMP on there. We're taking ATP, hydrolyzing it, putting an AMP on there. That's going to give us pyrophosphate as well, PPI, and that's going to dissociate. This process of going from ATP to AMP is what we're going to call using 2 ATP equivalents. He mentioned this in lecture, but you can take AMP and use ATP to get 2 ADPs. Bit hard to follow, but again, the general idea is that this is equivalent to 2 ATP hydrolysis events energetically. And that's going to come into account when we do a little bit more energy accounting work. So what's going to happen is coenzyme A is going to attack. Again, the AMP that we added on is a great leaving group, so coenzyme A is going to attack. AMP is going to hop off, and we have fatty acyl-CoA. That's our activated fatty acid, which we can then use to break down. Fatty acyl-CoA has a thioester linkage. We've talked about these in past lectures, but that's just a super energetically efficient way to store energy. So we've activated it. The next thing that we need to do is transport these fatty acids for breakdown. Right now we're in the cytosol. We need to be in the mitochondrial matrix for those breakdown steps. So So the way that we're going to move these around is by turning fatty acyl-CoA into acyl-carnitine. All acyl-carnitine is you're taking the CoA of fatty acyl-CoA and you're replacing it with this thing called carnitine. The reason we're doing that is that fatty acyl-CoA can't move across membranes. This is sort of similar to what we talked about in past lectures about not wanting to move NADH and FADH2 across membranes. You want to maintain those pools of cofactors within the cell, within different cells, so you don't want to specifically move that coenzyme A molecule across. So what we're going to do is we're going to take fatty acid, our fatty acyl-CoA, replace the CoA with carnitine. That's done with carnitine acyltransferase 1, or CAT1. Acylcarnitine is then going to move across the membrane. Once we get across the membrane and into the mitochondrial matrix, we're going to reattach the coenzyme A, throw off the carnitine. That's done by CAT2. And now we have our fatty acyl-CoA inside the matrix, and we can break it down. There's some thinking questions on this slide as well, on some other slides as well. Useful for review, useful to think through. Yeah. All right, fatty acid catabolism. We're finally at the point where we can break this down. The thing that is new in this process is beta-oxidation. We're taking that long acyl chain and we're breaking it down into acetyl-CoA. These other two things, oxidation and electron transfer. Electron transfer we have seen before, that's just TCA and the electron transport chain. So all that we're doing is we're taking our long chain, we're making it acetyl-CoA, and then the systems that we already have in place will break that down further and get us some energy, which is great. Beta oxidation is going to happen in 4 steps. It's going to be oxidation, hydration, oxidation, and then cleavage. You have seen that pattern before, sort of, with the end of TCA. When you go from succinate to fumarate to malate to oxaloacetate. And we're going to do the opposite again later. We'll get into more details about that later, but important to know the details of these steps. This is an oxidation step, this first step. Again, we want to get energy out of these molecules, so we're oxidizing. We're reducing FAD into FADH2. So we get an FADH2 out of that, which we can then send over to the electron transport chain. The next step is a hydration, so we're going to introduce water, and that's going to form this hydroxyl group, removing that double bond that we made in the previous oxidation step. And that's useful just so that we can do another oxidation step, which is step 3. Oxidation again. You want to get as much energy out of this as possible, so we are oxidizing our molecule. We are reducing NAD plus into NADH, which again we can send over to the electron transport chain and make some ATP. And the final step then is we are going to take this chain that we have and we are going to split it up. We're going to use the enzyme called thiolase. That's going to allow coenzyme A to attack. It's going to attack sort of right in here. And what we're going to do is we're going to split our, for this example, it's a 16 carbon molecule. We're going to split it into a 14 carbon molecule and acetyl-CoA. We can then send that acetyl-CoA into TCA, get some energy out of it. And we can do the same process again and again and again and again with this now 14 carbon long molecule. Okay, this slide has a ton of information, numbers, things like that. For me, it was helpful to sort of generalize. I think that it's very much possible on the test that they give you a question that's like, how much ATP do you, or how many ATP equivalents do you get out of breaking down a 14 carbon fatty acid, a 12 carbon fatty acid, things like that. These are some formulas, if it helps you think through that. We'll walk through this with the example in this picture. That's palmitoyl-CoA. That is 16 carbons long. I don't know why that. Oh, that's a beta, the box, beta oxidation. But the first thing to keep in mind is that we have a 16 carbon molecule, but we are not doing the cycle 8 times. We're doing the cycle 7 times because the final step is going to yield 2 acetyl-CoAs. So the number of cofactors, these reduced cofactors that you get, FADH2 and NADH, is going to be 1 less than the number of acetyl-CoAs that you generate. Important to keep that in mind, not get tripped up. If you have 8 acetyl-CoAs, which you get out of palmitoyl-CoA, you're going to get 7 FADH2s and 7 NADHs. You're then going to send those FADH2s and NADHs into TCA, the electron transport chain. We won't go into details of that, but again, for review, FADH2 For every one of those, you're going to get 1.5 ATPs. For every NADH, you're going to get 2.5 ATPs. So in total, you're going to get this many ATPs. I believe that's 108 for the breakdown of palmitoyl-CoA. There's one more thing to keep in mind, though. If you remember a long time ago when we were activating our fatty acid chain, we used 2 ATP equivalents. So you have to subtract 2 more. So for this example, you're going to end up with 106 ATP equivalents from your 16-carbon fatty acid chain, which is a lot. There's a bunch of advantages to storing fats. You don't need to know the specifics of this, the numbers of this, but it's important to know that fatty acid breakdown yields more ATP per carbon than cellular respiration. Fatty acid breakdown is something like 6.625 ATPs per carbon, whereas cellular respiration is about 5 to 5.3 ATPs per carbon. So this is more energetically efficient. The other advantage of this is that we store glucose in the form of glycogen. Storing glycogen involves a high amount of water because glycogen needs to be stored in a hydrophilic environment. This adds a bunch of weight and other stuff that you need to carry around. But fatty acid storage is pretty efficient because you can just pop it into a hydrophobic area and you don't need any other materials to store it. Cool. So a couple problems that we run into in beta oxidation. The first one is fatty acids with double bonds. The equipment that we just talked about, the enzymes, don't have the tools to break down a fatty acid that has a double bond. So our solution to that, again, we're going to take this molecule that we don't know and we're going to put it into a state that we know how to deal with. So we're going to take that double bond, isomerize, and we're going to move that double bond here. If you remember, that's the result of the first step of beta oxidation. So we can just do the next 3 steps of beta oxidation to cleave that and to get energy out of that. So we bypass that first step. So for each double bond, there's going to be 1 less FADH2 produced. You're going to get a little bit less energy. The second problem that we might run into is fatty acids with multiple double bonds. So this diagram is a bit confusing. Don't need to know the specifics, but it is useful to know the processes that we're using to solve these problems. So the first problem that we have, if we're looking at this 9 to 10 double bond up here, first we're just going to break this down. We're going to get as much out of it as we can, then we get to this place where that 9 to 10 double bond is between carbons 3 and 4. So all we're going to do is what we did in that previous slide. We're going to isomerize. We're going to move that double bond to a place where we know how to deal with it, which is right here. Oops. Sorry about that. It's going to run through the second, third, and fourth steps of beta oxidation. We're going to get an acetyl-CoA out. And now we are in this place where we've done the first step of this beta oxidation cycle, and our equipment doesn't have the ability to recognize this structure with 2 double bonds sort of back to back. That's this problem 2. You have double bonds between carbons 2 and 3 and carbons 4 and 5. So our solution for that is that we are going to reduce the first bond, the trans bond, and then we are going to isomerize as we did before. So we're just going to sort of get rid of this bond here, and then we're going to move that cis bond over to the trans configuration where, again, we know how to deal with it. That's over here. Sorry, I circled the wrong thing. Yeah. I know that's a bit confusing. Don't need to know all the enzymes in this, everything like that, but useful to know our solutions to dealing with this problem.
The only few enzymes that you have to know for this diagram is just the dienyl-CoA reductase, but that just means dienyl, you take your 2 double bonds and you collapse them into 1. And a good rule of thumb, the way I made sure to never mess this up, is I would literally do the alpha, beta, gamma, and delta carbons, so 1, 2, 3, 4, have them labeled all the time. And so that way I know exactly if I need that dienyl-CoA reductase, so the special enzyme, or if I just need just the simple one, which is just the enyl-CoA, which is just the 1, and you turn it from cis to trans.
Always label your carbons. It's very useful.
Yes, it does.
The last problem that we can run into is these odd-numbered fatty acid chains. This is a bit easier to deal with. We're going to cleave it as much as we can, and then we're going to get this sort of 3-carbon structure. All that we're going to do is we're going to carboxylate. We're going to add a carbon onto that. That's going to involve an enzyme that uses biotin. Biotin, again, is a sort of cofactor that we have seen before, used for throwing carbons onto things. So we're going to take that 3-carbon chain and turn it into a 4-carbon chain. The next thing that we're going to do is turn it from D to L. We're going to do the same thing we did before. We're going to oxidize to get rid of stereochemistry, and we're going to re-reduce, introducing the stereochemistry that we are interested in. And the final step is isomerization. We are just going to take this L-methylmalonyl-CoA that we have, and we are going to turn it into succinyl-CoA. An important cofactor for this reaction is coenzyme B12, sort of similar to vitamin B12. Important note, useful to know. Okay, another thing that we can do with acetyl-CoA instead of throwing it into TCA is this lovely thing called ketogenesis. Ketogenesis is the production of ketone bodies. These can be used by the muscle or by the brain to generate energy. This is sort of a way of packaging acetyl-CoAs that we have in the liver and transporting them around to areas that might be in need of more energy. These are the 3 ketone bodies that we have. There's acetone, acetoacetate, and beta-hydroxybutyrate. The steps of ketogenesis, also a bit confusing, but bear with me. The first thing that we're going to do is take 2 acetyl-CoAs, and we're going to condense them into acetoacetyl-CoA. We're going to get rid of a coenzyme A. It's just 2 acetyl-CoAs tacked onto each other with 1 less CoA. The next thing is we're going to throw another acetyl-CoA on there. That's going to get us beta-hydroxy-beta-methyl-gluteryl-CoA, HMG-CoA. Don't need to know the full name. You're going to take HMG-CoA and you're going to throw off an acetyl-CoA, and that's going to give you acetoacetate. Acetoacetate is sort of a final product. It's one of our 3 ketone bodies that we talked about earlier. You can take acetoacetate and turn it into D-beta-hydroxybutyrate if you would like to. Acetoacetate will also form acetone. Acetone is not nearly as useful as the other 2 ketone bodies because it is a waste product. You can't get any energy out of it, and you can't process it any further. So the other 2 are the ones we care about, acetoacetate and beta-hydroxybutyrate. Now, once you take those, you package them up, you send them over to the brain or to the muscle cells. The next step is to break them down. Ketone body metabolism is going to be done, again, mitochondria of muscle and brain cells. We do this when we don't have as much activity in the TCA cycle or if we have less intermediates. The first step of that is we're going to take beta-hydroxybutyrate, if that's our ketone body, and turn it into acetoacetate. If we just start with acetoacetate, we don't have to do that step, obviously. The next step is a sort of activation step. We're going to turn acetoacetate into that intermediate of ketogenesis that we saw before, that is acetoacetyl-CoA, just an activated form. The last thing we do is take acetoacetyl-CoA and we're going to chop it up. We're going to introduce a coenzyme A, that same thiolase enzyme that we saw earlier with the final step of beta-oxidation. And we are going to cleave acetoacetyl-CoA into 2 acetyl-CoA molecules, which we can then put into TCA. So it's important to know not only what ketogenesis and ketone body metabolism are, but also when they occur. So when we're going to be doing this is when the body is in a state of starvation. So if you're in a state of starvation, you're going to have less glucose. If you have less glucose, the body is going to be more gluconeogenesis. As a result, remember, gluconeogenesis takes oxaloacetate, that's right here, and uses that to turn it into glucose. Because we're taking oxaloacetate and we're still running that cycle, we're going to have a depletion of TCA intermediates. The TCA cycle is going to slow down. You're going to have a bunch of acetyl-CoA that accumulates. And because of that, acetyl-CoA is able to become acetoacetate, which is then used in ketogenesis. Okay, one of the last pathways that we're going to talk about. We're almost there. Well, from my end, then you get to deal with amino acids.
Yeah.
Sorry, guys. We're going to talk about lipid biosynthesis. So we talked about breaking these lipids. Yeah, sorry.
I have a ketone bodies question.
Yeah.
If it breaks down to acetyl-CoA, how is it useful for energy? Or like, just because we talked a lot about how like putting acetyl-CoA in TCA doesn't replenish it.
Yeah, that's a great question. I don't know. Do you know?
In terms of acetyl-CoA.
Yeah. I think it's, this is a confusing thing because we did talk a lot about that. But for the sake of this, just something?
I think because we're not necessarily not breathing. Does that make sense? So like aerobic respiration is still occurring. So the acetyl-CoA goes through TCA cycles. We're trying to exhaust all of the possible energy that we have. By this point, we don't have glucose, so obviously it's not going to occur efficiently. And we're left with acetyl-CoAs because that's the last thing we have. Our body's trying to survive somehow. And so acetyl-CoA will go through TCA cycle, essentially, and keep doing the cycle, and it'll just take in like the 3 NADHs, the 1 FADH, and then form the ATPs from that. So that's like a last resort. It's definitely not efficient, you're right. But that's the way we would use acetyl-CoA. It's like a last resort for this type of thing. And aerobic respiration would still be occurring because we are breathing.
Yeah, we talked a lot about how acetyl-CoAs, like the carbons from acetyl-CoA, are sort of just thrown out as CO2. But acetyl-CoA is an intermediate. Some intermediate is better than nothing. We're going to sort of get those cycles going as much as possible. That is confusing, though. That's a good point. Okay, lipid biosynthesis. We're sort of just going to reverse all the steps that we did in beta-oxidation. We're going to use different ingredients to do that. Our carbon donor is going to be malonyl-CoA instead of acetyl-CoA. Malonyl-CoA is a 3-carbon structure. We'll talk about why that is later. But instead of acetyl-CoA as our acyl carrier, instead of coenzyme A as our acyl carrier, we're going to use ACP, acyl carrier protein. Of course, this time we are using energy instead of getting energy, so you're going to have an electron donor instead of an electron acceptor, and we are doing this in a different place. This chart is great to have. I think there was a test on this question on the test last year as well. But useful to think about how these pathways are related. In practice, they're very similar, just reversed and with different pieces. So the first step is we have to get to the cytosol. We are using acetyl-CoA. A lot of that is going to be in the mitochondrial matrix. We want to take it out and go into the cytosol. The way that we're going to do that is with this sort of shuttle system. So we're going to turn oxaloacetate into citrate by loading our sort of acetyl group onto that. This is, again, the first step of TCA, if you remember. When we get acetyl-CoA out of glycolysis and we throw it into TCA, we're taking oxaloacetate, giving it acetyl-CoA to form citrate. We have specific transporters that allow citrate to diffuse across the membrane. Citrate is going to go into the cytosol, and from there we can extract that acetyl-CoA, which we can then use for fatty acid synthesis. There's 2 different sort of return pathways to this cycle. The first one is oxaloacetate. You reduce it into malate, and then you shuttle it through. This is similar to the transporters that we have in the malate aspartate shuttle. That doesn't really do anything for you energetically. The other pathway that we have is that cytosolic NADH, NADPH specifically, is extremely important for fatty acid biosynthesis. So if you need more cytosolic NADPH, the body's going to recognize that, and it's going to do this bottom pathway. You're going to take malate, you're going to turn it into pyruvate. That is an oxidation step. You're reducing NADP plus to form NADPH, which you can then use. Pyruvate will then transport through the membrane, turn it into oxaloacetate, can do the cycle again. Important thing about that is 2 different pathways depending on the energetic needs of the cell. Okay, making malonyl-CoA. This diagram was shown in lecture, the leftmost one. The details of that are not that important this year. A couple things to note is that we are making malonyl-CoA with the enzyme acetyl-CoA carboxylase. That tells you a little bit about what we're doing. We're taking acetyl-CoA, we're sticking CO2 onto it. That's going to use the biotin cofactor, which again, we've seen so many times. This is the commitment step of this process. It's extremely heavily regulated. That diagram on the bottom middle is sort of a memorizing heavy step, but you can also sort of intuit your way through it. So if you have high levels of insulin, that means you have the body has a high level of energy essentially. So you can do that step of citrate to acetyl-CoA to malonyl-CoA more readily. You can make stuff. Whereas on the other hand, if the body is short on energy or has a high demand for energy, that's if you have glucagon or epinephrine, you don't want to do that pathway because you want to take the energy that you have and use it for those processes. We're going to synthesize fatty acids with this big enzyme complex called fatty acid synthase. This has a bunch of different components. Important thing to know, we're going to do the reverse of what we did in beta-oxidation with this enzyme, and also we are going to use ACP, acyl carrier protein. This is what Professor Solomon described as a tethered version of coenzyme A, just to move stuff around. And let's get into the steps of it. So the first thing that we're going to do is we're going to load that synthase enzyme with acetyl-CoA. We're just doing that the first time, though. If you have a long chain, acetyl-CoA is just used to start this process off. You're also going to load malonyl-CoA onto that. What we're going to do is the CO2, the reason that we want malonyl-CoA instead of acetyl-CoA is that the CO2 on malonyl-CoA is going to act as a good leaving group. So when we have CO2 on malonyl-CoA, that allows for a condensation reaction where we can stick those two together. CO2 is going to leave. We end up with a 4-carbon molecule. The next step, again, we're doing the reverse of what we did in fatty acid metabolism. What we did there was oxidation, hydration, oxidation, cleaving. What we're doing here is condensing, the opposite of cleaving. Then we're going to reduce, that's the opposite of oxidation, obviously. Going to require energy to build these fatty acids. Going to do a dehydration step, and then lastly, another reduction step. The result of that is this 4-carbon long chain. We're going to move this chain over to the other site. We can put another malonyl-CoA in if we want to increase the length of this chain, and do it again, as much as we need. In general, the body will make palmitoyl-CoA, palmitate, which is that 16-carbon chain that we talked about earlier. The final step of this process is once you've made the chain as long as you want it to be, you are going to use a thioesterase, and that's going to cleave the palmitate off of the thioester, and now you're just going to have that free 16-carbon fatty acid. You can do what you want with it. You touched on this briefly in lecture. This diagram is not something you need to memorize. I think it's just useful to know the different options that you have here. You can use that palmitate directly. You can break it down into energy if you need to. You can lengthen the chains. That allows you to store more energy in one molecule. Also, longer chains are useful for impacting membrane fluidity, which we talked about earlier. The other option is adding double bonds. Again, changes membrane fluidity, depends on what you need from your lipid. That step is done with the enzyme fatty acyl-CoA desaturase. Again, details of that are not important, just useful to know that these are the different paths that you can take. All right, a couple more synthesis pathways, and then I'm done here. You'll stop hearing my voice. So the first thing that we're going to talk about is synthesizing triacylglycerols and glycerophospholipids. This process is pretty similar. The first thing that we're going to do is make that backbone. What we're going to do, again, the backbone is glycerol. What we're going to do is we're going to take glucose, sort of the opposite of what we did before, turn it into glyceraldehyde 3-phosphate. That's just a step of glycolysis. And then again, we need to invert the stereochemistry. Glyceraldehyde 3-phosphate, the one that we have right now, is in the D configuration, but we need it to be in the L configuration. So what we're going to do is oxidize it to get rid of the stereochemistry, reduce it in a way that we want to invert that stereochemistry. Another pathway, that's this right one here in this picture, is that if you have a glycerol already, you can just throw a phosphate onto it, and you have glycerol 3-phosphate. The second step of this happens both for triacylglycerols and glycerophospholipids, is we're going to want to add fatty acids. The details of this are not that important, but you're going to take 2 ATP equivalents. You can use the enzyme acyltransferase, and you're going to add the 2 fatty acids onto your glycerol backbone. The last step of this is if you have a glycerophospholipid, you want to add a head group. If you have a triacylglycerol, you can just do that same reaction as before, add on another lipid and call it a day. In this case, you want to add a head group. So what you're going to do is you're going to either activate your backbone that you've been making, or you're going to activate the head group that you want to throw onto that. You don't need to know the specifics of these reactions, is what Professor Solomon said in lecture, but it is useful to know that there are 2 different ways to do this, and also we are activating by adding a nucleotide specifically. We'll talk a little bit more about the reactivity of nucleotides in one of the later lectures. All right, sphingolipids. All you need to know about this is that we're going to use serine, as you can see in this box here. Serine amino acid, and we're going to sort of make some other modifications. We're going to throw a fatty acid on there, throw the head group on there in the way that we did before, activating either the backbone or the head group itself. don't need to know the details of this image. It was included in the lecture for reference as well, so useful to see, but you don't need to memorize it. Cholesterol. I think this is the last synthesis that you'll hear from me. This happens in a bunch of steps. The first thing that we're going to do is we're going to take acetate. You're going to take two acetates, you're going to combine them together into mevalonate. That is then activated. You're going to take mevalonate, the activated mevalonate, you're going to cleave it into two activated isoprenes. These are 5 carbon molecules. These are the building blocks of your cholesterol molecules. You're going to combine 3 of these activated isoprenes, and that's going to form one, that's a typo, 15 carbon molecule. You have to do that twice. You need 2 15 carbon molecules. You're going to combine those 2 molecules to form squalene. Squalene is what you are going to cyclize to form cholesterol. I think this is a useful way to think about, to do some sort of carbon accounting. You have 2 5 carbon molecules. That's your activated isoprenes. Those are going to combine into a 10 carbon molecule. You're going to take another 5 carbon activated isoprene to form a 15 carbon molecule. Take 2 of those, combine it, and you have 30 carbon squalene, and you cyclize it. All right, the last thing here is regulating cholesterol metabolism. Regulation is important in pretty much every unit that we've talked about. It's going to be important here as well. This has to do, again, with the energy level of the body. So if you have high levels of insulin, you have a lot of fuel in the body, so you can do more synthesis. You can make a bunch of cholesterol if you need to. Again, this is the step that's regulated. On the other hand, if you have low levels of energy in the body, you're going to have glucagon. Glucagon signals low fuel availability, so there's going to be less cholesterol synthesis that is going on. He also mentioned these statin medications, the way that statins work. They're used to combat high cholesterol. Statins work by inhibiting this HMG-CoA enzyme, HMG-CoA reductase, and what inhibiting that step is going to do is you're going to accelerate this export step right here. If you accelerate the export step, you're just taking out all of that sort of bad cholesterol from the blood, which allows you to cycle in more cholesterol that is good as needed. Statins work by acting as a competitive inhibitor for HMG-CoA reductase. They mimic the structure of mevalonate.
Question?
Yeah.
Question?
How does like the cholesterol esters, like how does that remove it from the blood? Like how does that actually work?
The which one, sorry?
The cholesterol esters,
Step here.
like what is that like actually like remove from the blood? Like, how does that work?
What do you mean by remove from the blood?
Because like cholesterol is being like removed from the blood to turn into the ester form, right?
Mhm.
So like how does that actually like work?
That's a good question.
So cholesterol esters have an ester group, which we know is polar, right? And so we know that cholesterol can freely move in and out of the cell itself because it can pass the plasma membrane. And you would attach this ester group to it to keep it trapped inside the cell. It's very similar in the sense of like glucose and glucose 6-phosphate, how glucose 6-phosphate keeps it inside and doesn't just travel between. So it's a similar case here, or alternatively, it'll keep it out in the blood as well. And it interacts with specific apolipoproteins, but that's beyond this course.
Because we're doing that in the cell, we're sort of trapping it in the cell, like Alex said. The last thing here that I forgot to mention is that cholesterol is also going to regulate itself. What it's going to do is it's going to either inhibit that step up there, HMG-CoA reductase, or it can also inhibit the intake of cholesterol, LDL cholesterol specifically, on the extracellular side. So when you have high levels of cholesterol, you're going to stop making it, and you're also going to stop taking it in. It's just going to sort of float around. Any questions on lectures 13 and 14? Awesome. These are just some sort of tips to review. The slides are posted again. These lectures are pretty memorization heavy, so take your time with the review. Let us know if you have questions. I hand it over to you.
Yeah. Use the practice questions to guide exactly how you should be applying the stuff you're memorizing, because I think that's the biggest thing is it feels like it's a lot and you don't know how to necessarily apply it. We purposely try to put a lot of practice questions and then also show up to the Jeopardy because I'm going to be putting a lot of time into making some questions as well. I won't be there, but I'll be putting a lot of time into that.
It's so fun.
Oh, yeah. But yeah, that's just a bunch of questions that you guys can answer on your own time. So we'll go into lecture 15. I'm going to present this a little bit different than how Professor Solomon did. So this one, hopefully it makes a lot more sense as to why you're learning these different pathways. So I really wanted to start first with the objectives because I'm probably not going to reference them again, but it is good to keep us grounded. So the first thing I want to talk about is we're going to talk about the fate of these amino groups that are on the amino acids. We know that they're an ammonia group, they're NH4, and we know that's extremely toxic to our bodies. We don't want it in the blood, and so we want to remove it. We want to excrete it through our kidneys. And so we're going to talk about that first, specifically removing the ammonia from the body in a safe way. And that's the whole reason why we have the urea cycle. Then the second thing I want to talk about is what happens when we get rid of the ammonia groups and we have the carbon skeleton, right? That doesn't just like do whatever. There's a way of breaking it down into usable energy or into useful metabolites for other processes. So we'll talk about that as a second point. And then we'll pivot to amino acid biosynthesis. So let's say we are low on amino acids, or let's say we have high energy, right? And we can build things up. One of the things we may want to build up is amino acids. And so there are certain amino acids, not all 20, but like a certain few that we can build up. And I'm only going to highlight 5 specific pathways that you guys should know for sure, like have it memorized. But we'll go over that and then we'll go through the rest of the stuff. But I think that'll get us going for a little bit. So just want to emphasize with this diagram, I highlighted the boxes that you guys should always keep in mind when it comes to this lecture. So as you can see here, there's a pivot, or when we start at the very top, we said we have our amino acids that are in the body that need to be broken down. They're going to be broken down, as I said, the ammonia group and the carbon skeletons, and those are going to go through the respective pathways. With the ammonia group, we see that it's going to get incorporated into carbamyl phosphate, and that's going to go through the urea cycle. And the urea cycle is exactly how we get rid of ammonia from our body, because we can't just let ammonia transport through the blood. What we have to do is convert it into urea. So that's the whole point why the cycle is named that and why it's there in the first place. Is because ammonia is toxic in the blood. We have to convert it into a safe form, and then it'll get excreted. That's the whole point of that. And then in terms of the carbon skeletons, we make those alpha keto acids, right? But the alpha keto acids, if they are ketogenic, will form ketone bodies. And we know that ketone bodies are not glucogenic at all. The acetoacetate, the acetone, the acetyl-CoA, like all that stuff is ketogenic. Whereas the ones that are glucogenic are going to replenish our TCA intermediates right here. And then it's going to go, and usually the ultimate fate is that it makes oxaloacetate. And then we know that oxaloacetate can make glucose from a gluconeogenesis, which I already forgot already. But that's just like an overview. Keep this in mind as we go into the details now. So first thing I want to talk about is, let's say, forget about all the Cori cycle, forget about the transport in the blood. I first want to talk about in the liver, where we actually process this ammonia in the first place. So let's say we got these let's start with these blue rectangles, right? These are amino acids from different parts of the body. The top box is from things that we eat, the essential amino acids that we have to consume. The one on the right is alanine from the muscles. We know that skeletal muscles, they're going to transport alanine, and I'll go into specific details why it uses alanine. And then the third one is just from regular cells, they use the glutamine transport system. So those are the 3 different ways you can get amino acids into the liver. The liver is like the processing center for amino acids, for buildup or for breakdown. And I really want to emphasize as well that we're using a lot of transamination reactions, transferase reactions. And what that is we're shifting the amino groups from these amino acids that we've now brought into the liver cells, and now we're basically stripping them off. And so as you can see here, we end up forming our ammonia, and that gets incorporated into carbamyl phosphate. So that's like the output. And so that's why I really want to emphasize what is going on in the liver is we're taking everything from the body, all the ammonia, and we're processing it and putting it through and funneling it into the urea cycle itself. But before I go into the urea cycle, I want to talk now more about those blue rectangles specifically. So I know there's a lot on this slide. We'll go into it one by one. So the first thing is that in order for us to get these amino acids to go into the blood, we have to do, from the cell, we have to do these transamination reactions. So as you can see on this one right here on the top right corner, we have to convert alpha keto acid into glutamate, and then glutamate can be converted into glutamine, and that can transfer through the blood and get into the liver. So that's like the first thing I wanted to start off with is within these cells, we have to undergo this packaging before we put it into the blood, and it goes into the liver. We have to do some packaging. And then there are specific kinds, right? So the first one I want to talk about is the actin muscle. And this is known as the Cori cycle. It's this diagram right here on the bottom right. So what I wanted to emphasize is that muscles, they don't use glutamine, they don't use glutamate, they use alanine and pyruvate. Now, the reason why they use this is because muscles have to rely, and we know this, they rely on anaerobic respiration. And we know that if it relies on anaerobic respiration, what's going to end up happening is we're going to have a buildup of pyruvate. And what that pyruvate has to do is it'll become lactic acid and whatnot. But additionally, it can undergo this alanine aminotransferase reaction, where then we can incorporate a nitrogen onto it. We can incorporate a nitrogen group onto it, and it becomes alanine, and that gets shuttled to the liver. And the reason why you would see this process, this Cori cycle occur, is if like we're resting, we're not necessarily working out anymore. We're trying to build up the lactic acid that we built, or the alanine itself, or the pyruvate buildup that we've had. So that's what the Cori cycle is. That's one cell type. Now, in terms of the other one, which is just regular cells, what happens there is we're going to use the glutamine, as I mentioned. We know the difference between glutamate and glutamine is that glutamate has a carboxyl group on the R group, whereas glutamine has an amide. It has that extra nitrogen stored onto it. So again, we're going to use this transamination reaction. We're going to take ammonia from within the cells and attach it to the end of glutamate to form glutamine. Now we can transport it safely because we know glutamine is not charged like glutamate. Glutamate is negatively charged, and it can't pass through the cell layer, at least not without help of a transport protein, or an ion channel, sorry. But in this case, if we convert it to glutamine, it can now have a better time and have access to new transporters, to go into the blood and do its thing. And those are the two main methods that you guys should definitely know of moving these amino acids from tissues into the liver. Yes, okay. And then the last thing we need to do before I move on is that we need to form this free ammonia, inside the liver, right? What we have to do is undergo glutamate dehydrogenase reactions, which all that means is that we have to do an oxidation reduction reaction, and that'll basically pull away the ammonia. So now it can be freely used in the urea cycle. So this is like the final step where it converges, right? We have a convergence of the regular tissue, and we have a convergence of the skeletal tissue. And it gets processed, and the final step is this glutamate dehydrogenase, where we ultimately form the ammonia that can be used in the urea cycle. So I really want to emphasize that there is convergence inside the liver once you're there. Okay, so now we just formed this ammonia. What ends up happening in the liver? And just saying it out right now is the urea cycle. And we know that ammonia itself is toxic, urea is not. And so we have to do this conversion where we basically incorporate the ammonias into urea. And so there's a two-step process, right? Obviously, the urea cycle is a cycle, but I don't really want to go into the specifics because that's not worth our time. I want to go into more of the bigger picture. So the first thing we have to do is enter the urea cycle in the first place. How do we do that? We do that through step one by forming carbamyl phosphate. This is a new molecule that you guys haven't heard of in the previous pathways. And it's basically the rate-limiting step of the urea cycle. So there's probably going to be tight regulation in this, which I'm kind of jumping the gun and foreshadowing, but there's going to be tight regulation during this step. And what this is, without getting into the weeds, is we're going to use two equivalents of ATP, I'm pretty sure. Yep, one right here on the left, and then another one on step 3. We're going to use two equivalents of ATP, and that's going to allow us to incorporate our ammonia and form our carbamyl phosphate. That's basically all you have to know. You don't need to really know the mechanism itself. They're not going to ask you to draw it. And then step 2 is, let's say we do form that carbamyl phosphate, right? Well, now it's going to get incorporated into the urea cycle. And what ends up happening is carbamyl phosphate is going to combine with ornithine to form our citrulline. Now, I want to keep this in mind as we go through all these lectures is the spatial biology. What I mean specifically is that this step that I'm talking about with carbamyl phosphate and ornithine addition, it's happening in the mitochondria. It's not happening in the cytosol. So that's something to keep in mind as well while we're going through these lectures, because on the last year's exam, right, they asked a question where it's they list out a bunch of pathways, and it was just urea cycle, beta-oxidation, and then it said which compartments do these happen. And so then you give a answer choice of urea cycle happens in both, and that is an option. So you have to know the spatial biology with all the pathways, because they could ask a question on that. It was on last year's exam. And we'll go into depth. Do you guys really want to review the urea cycle, or do you think that it's worth skipping over, just keeping the bigger picture? I can go over it if you guys want. That's fine. But I'm going to integrate it into all the pathways. Okay, I'll keep going. I'll just keep the bigger picture because I don't think it's worth our time to spend on the little details. So I color coded a lot of these boxes. The one thing I want to start with is our inputs, right? So I really wanted to start with the glutamate. We see that the glutamate here, it's in a purple box. And I wanted to emphasize this because it's the same exact glutamate, where the amine group from the glutamate is being extracted and we end up forming our alpha keto acid, our alpha ketoglutarate. And this is from the same molecule again. So the alpha ketoglutarate is going to form and it's going to do its own thing, right? It's going to be part of this malate aspartate shuttle. Whereas we see here, the stripped ammonia is going to be incorporated into carbamyl phosphate. So we see how glutamate is not just only doing the urea cycle, it's doing both things at once simultaneously. So that's something to keep in mind as well. And then in terms of this, I want to highlight the yellow boxes. This is the energetics of the urea cycle. How many ATPs are consumed? For the urea cycle, for one rotation, it's going to consume 4 ATPs. There's 2 ATPs at the very beginning, as we mentioned before. And then when it comes to the citrulline right here, combining aspartate and citrulline, it's going to be converted into arginosuccinate. It's going to use 2 equivalents of ATP. And so I'll show here, but essentially we have our ATP right here, and it forms a pyrophosphate. And this is essentially used as 2 equivalents. And another way to think about it also, if that's a little bit confusing, is that let's say we have that AMP. How much is the delta G to get back to ATP, right? It's technically the 2 equivalents of the phosphate groups to be attached to it. That's another way you can think about the equivalence thing.
And we saw that before. Exact same thing as activating our fatty acids.
Yes.
Throwing an AMP on there.
Yeah. So once we form this arginosuccinate, what's going to end up happening is it's a large molecule. It's 6 carbons, as you guys know, right here or not even 6. I don't even know how much, but it's a large molecule essentially. And we're going to split it up. We're going to form a fumarate, and we're going to form an arginine. The arginine we know is part of the urea cycle. The fumarate, that we form is going to be part of this new shuttle that we haven't seen, and it is a part of the malate aspartate shuttle. This shunt is called the aspartate argininosuccinate shunt of the citric acid cycle. All that is like an extra addition. And this fumarate, you don't need to know the specifics, can be incorporated into the malate aspartate shuttle. And this is what allows us, again, to regenerate the NADHs. And the NADHs are important because we need to be able to reduce the cofactors and whatnot. I think I actually have a point here. Let me see if I'm not. Oh, yeah, that's what it was. Sorry. So the NADH is important. There's a reason why. It's because that last step that we talked about with this liver processing to convert this NH4, we're going to need the NADPH, and we need it inside the mitochondria. So we're going to need this malate aspartate shuttle to work in order for us to do that stripping of the ammonia from the glutamate itself. Hopefully that makes sense, and hopefully I didn't confuse, but it's in the slides, and you can go through it slowly on your own time. Any other questions so far before I move on? Okay. Yeah, I think that's most of this. Yeah. Okay. And then remember how I talked about the rate limiting step in the urea cycle? It was that the formation of carbamyl phosphate. So this is going to be heavily regulated in 2 ways. There's a direct activation and indirect activation. The direct activation has to do with our N-acetylglutamate, where it will directly stimulate carbamyl phosphate synthetase 1, right? This carbamyl phosphate synthetase 1 is the thing that actually incorporates the ammonia with our bicarbonate to form the carbamyl phosphate. That's the first thing. That's the direct stimulation of it. Now, the second thing is that acetyl-CoA can combine with glutamate to form this N-acetylglutamate, and that can be stimulated by arginine. Now, why does arginine stimulate it? Well, in the urea cycle, arginine is one of the metabolites that forms when we break down an arginosuccinate. So if we have a surplus of this, we want the urea cycle to keep going. So that's exactly why arginine would stimulate it. So those are the 2 different types of regulation. There's a direct one where, again, it acts directly on the enzyme, or the indirect where it will stimulate the activator, if that makes sense, the formation of it. Okay. And so let's not forget, we've been talking about ammonia this entire time. That was the urea cycle. That was this malate aspartate shuttle. Now let's focus more so on the alpha keto acids, these carbon skeletons. And just to orientate ourselves on this right diagram right here, I noted that the color coding is reversed. So ketogenic shows orange here, but on the diagram that you guys have in your lectures, it's blue. So like, don't get that confused. I'm sorry. They just didn't overlap when I looked it up online. But the best way to memorize in terms of whether it's ketogenic or glucogenic is first memorize the only ketogenic. So this pink box, memorize that it's lysine and leucine that are only ketogenic. And this could be a question where they say, oh, what is leucine glucogenic or ketogenic? And then you would know from this memorization. And it's also good for the MCAT as well. They will expect you to have this memorized too. So that's the way I memorize it, is first memorize the ketogenic, then memorize the overlaps, the ones that could potentially be glucogenic and ketogenic. If you memorize these, you don't need to, technically you don't need to memorize these other ones like gluconeogenic, because you know which ones are solely ketogenic and which ones overlap. And then by that definition, you know which ones are only glucogenic. And then we'll go specifically into each specific pathway, and I made it sure to like separate them and make them easier to understand. But we'll move on to the first one, which is this very complicated one. So all these pictures, I just kept them here for your reference. We're not even gonna like bother going through the mechanism, all the enzymes. The most important thing is that there's technically like two pathways within this. So the first pathway you can consider as our ketogenic pathway. It's this one on the left right here, on the bottom left, where threonine can become acetyl-CoA. And so just kind of orientating ourselves to like the scheme, whatever, on this middle diagram, we see that we have threonine in this box, right? And what ends up happening is that our group from threonine gets converted into acetyl-CoA. That's the ketogenic part of threonine. So if they ever ask you, like, how can threonine be ketogenic versus glucogenic? We know that the R group is acetyl-CoA, and that's ketogenic. And then the backbone, this carboxylate and ammonium group, or amine group, sorry, that's going to be our glucogenic, right? And so what ends up happening is the R group becomes acetyl-CoA. What happens to the backbone? The backbone becomes glycine through this reaction. So the pathway is like threonine to glycine. And then what happens with the glycine is it can be combined with, I believe it's methylene. And when that gets combined, we form serine. And the reason why in the heading I color-coded these with the same colors, because it's the same exact serine that's going to feed into pathway 2. So pathway 2 has three little like ways that these amino acids could converge on pyruvate, right? So we have the serine, which I think that it is important for you guys to know serine dehydratase. It is important for you guys to know like the alanine aminotransferase, like basically the enzymes that directly get you to pyruvate. Those are really good to know because they could ask you to draw like what's the final like enzyme that does that conversion. But besides the point, going more towards the bigger picture, so the serine goes into pyruvate. And remember, the serine originally had to come from the backbone from threonine as well, from the glycine. So it's almost like a sequential pathway, and it ultimately leads to pyruvate. And then we also know that tryptophan and alanine, they're in the same pathway, and it ultimately leads to pyruvate. And then the last one is just cysteine, which also ends into pyruvate. So this is like the two pathways. I decided to separate them because it makes it easier to understand what's the ketogenic side and what's the glucogenic side, and why threonine is able to do both. But they examine this very in depth. But does anyone have questions about this pathway? Yeah.
Can you explain what glycine's role is?
Yeah, so glycine is the backbone, right? And what ends up happening is it's going to get added to it. What can happen is the glycine can be added to the methylene, right? So this is technically glycine. It can go two ways. It can go the carbon dioxide way, which we don't really care, it just forms carbon dioxide, or the more important way, which is the methylene is I believe it's a C. I don't necessarily know the functional group. You can look it up online, but methylene has carbons inside of them. And so we attach them to the glycine and to the R group, and that ends up forming the serine. Does that make sense? Okay. Any more questions? Okay. So moving on to the next pathway, I changed the boxes down below where we're going to talk about tyrosine and phenylalanine. These are the other two aromatic amino acids. So remember, tryptophan was incorporated into this pathway. So now we're going to talk about the last two. So tyrosine and phenylalanine. We know that they're both glucogenic and also ketogenic. They have both those properties. So the one thing I wanted to emphasize is that realistically, the only thing you need to memorize are two things. The hydroxylase catalyzes the conversion of phenylalanine to tyrosine. So that is always going to be true, is like phenylalanine will go into tyrosine. And then through a bunch of dioxygenases, which is just some random enzyme. Just memorize the name of the enzyme itself. You don't need to memorize all the specific steps. Just know that through dioxygenases, the tyrosine gets converted into both fumarate and acetoacetyl-CoA. And the former is going to be glucogenic. The latter is going to be ketogenic. So basically, because like tyrosine is such a large molecule, it breaks down into both these substrates. So same thing with phenylalanine. It's a large molecule, right? So when we split it apart, the R group or whatever it is, because you don't even need to know that specific detail, you just have to know it's large enough that it'll split into both the fumarate and acetoacetyl-CoA. So it's both glucogenic and ketogenic. Does anyone have questions on this pathway specifically? Okay. Moving on to the next pathway. This is the branch. Oh, sorry.
Yeah, we need to know about the, I was like looking at these yesterday, and there's like a bunch of the, like Solomon talks about like the clinical applications of like the inhibitions of some enzymes will like be things like PKU, and then there's like 4 other like, of like these diseases that are like caused by the enzyme inhibition or like not being effective. Like, are we expected to know like all the enzymes and how they like relate to a certain like tyrosinemia 2, 3, PKU? Like, what do you think we should be knowing about that?
No, because I did look at your guys' slides. I will say, like, I'll explain. The scapegoat answer is just say anything on the slides will be tested, but I want to say that it's probably not worth your time if you're still reviewing a lot of the bigger picture concepts to not focus on that. That's going to be what, like worth 2-4 points. So like it's, I would say that if you have time, definitely do it, but you don't have to necessarily know like, oh, phenylalanine hydroxylase inhibition is going to lead to PKU. You just have to know down the pathway it could lead to one of these clinical applications. Does anyone else have any questions on this? Okay. So moving on to pathway 4. These are the branched amino acids. So we have valine, isoleucine, leucine. And if I were to show you guys the amino acid structures, we know valine is the 3-carbon R group. We know isoleucine is the one on carbon 2 where it's branched. And then leucine is the one where carbon 3 is branched as well, or carbon 2, whatever it is. But they're all branched. And the bigger picture is that we're going to convert our branched chain amino acids and use this aminotransferase, attach an amine group, and then we're going to get rid of the amine group, We're going to form our alpha keto acids. And then we're going to use a dehydrogenase to convert them into their acyl-CoA derivatives. And that's just going to get converted into either succinyl-CoA, it's going to get converted to acetyl-CoA, or the acetoacetyl-CoA. Now, the best way to memorize which ones are going to go to which substrate is we know that leucine is the only ketogenic one, right? On this diagram, we know leucine is the one that's exclusively ketogenic. So by this logic, like we would, and you can like reference this diagram and try to like ingrain it into your head, but leucine is going to be on the bottom, and because the bottom two options are because the bottom two options are acetyl-CoA and acetoacetyl-CoA, those are ketogenic. They do not become glucose because we know the acetoacetyl-CoA will get broken down into 2 acetyl-CoAs, and we know that doesn't get converted into glucose at all. So we know that the leucine is the only one that will form the 2 ketogenic substrates, whereas the other 2 options, isoleucine and valine, specifically the isoleucine, can form both the ketogenic and the glucogenic. And then valine, once you memorize the middle part of this Venn diagram and then the left part, you'll know which ones are glucogenic. So then by that memorization, you'll also have, okay, valine, we know it's not in the middle, so it's going to be exclusively glucogenic, which is what we see here. It forms succinyl-CoA. So if you were memorizing this, you don't really have to know all this. I mean, honestly, you could, if you really have the time, you could memorize it, but this is like such low yield that it's better to spend more time on the bigger picture. You're going to be tested for sure. Like I would be 95% confident on breaking down the fatty acid chain and calculating how much ATP you're going to generate from like a 16 carbon, 18 carbon, or 20. They may ask you how to do that. That's way more likely than them asking you, what is maple syrup disease? What is the mechanism of action? Like it's not conducive to you guys' learning. Does anyone have questions in terms of pathway 4? Okay, and then this is the last pathway. It's a lot more simple. It's just the conversion of asparagine and aspartate, and it's going to get broken down into oxaloacetate. And we know that this is a glucogenic, and so we see that this one is pretty easy to memorize, all like the specific details. Asparagine is going to get converted into aspartate, and the relationship between asparagine and aspartate is that asparagine has that amide, the aspartate has the carboxyl group, so we're going to get rid of the ammonium group. So we're going to use a asparaginase, which just gets rid of the ammonium and replaces it with the water. And then from there, we're going to do an aspartate aminotransferase to form oxaloacetate. A good thing to know is that oxaloacetate is the alpha keto acid of aspartate. That's a very good relationship to have in mind and like almost memorize and be able to intuit. Because they could ask you like, oh, what's the alpha keto acid of aspartate? And you just have to kind of like know it or have it memorized. Does anyone have questions about pathway 5? This one was a little bit more straightforward. Okay. And then another thing that I want to talk about is 5 important cofactors, not just for this lecture, but just all the lectures combined. So the first one is PLP. It's part of a transamination reaction, and actually as we go further on onto lecture 17, the nucleotides, it's also a decarboxylase as well. That is one thing to note for one of the cofactors. Biotin we've already talked about extensively, which is just carboxylation, carbon dioxide addition, and we have to use ATP to do so. Adomet is also a 1 carbon transfer, but this one is a methyl group. So it's going to do some crazy radical reaction. All you need to know is that it just adds a methyl group to whatever the substrate is. Then we have THF, which is just a 1 carbon transfer where, again, I'm pretty sure THF, you just add a methyl group as well, but I need to double check that for sure. And then THB is summit. That one I've seen before, but it's not really tested that much, so I wouldn't like worry too much about it. I can't recall off the top of my head which one it is. I will say they may ask you to know what the cofactors look like. It is good to like take pictures, upload them to Quizlet, and simply just like get rid of the name, and then try to just recognize them. It's not that you're trying to draw them, it's the fact that you can recognize what it is. And that's one of the easiest ways that they can test you on this exam is they'll have pictures printed of it, and you just have to write what that cofactor is. Does anyone have questions on that? Okay. So that was all amino acid catabolism. Does anyone have questions about like this overall thing? If not, I can move on to the other stuff as well. Okay. Is there a question? So next thing we're going to move on is to amino acid biosynthesis. I want to note that we can't make all 20 amino acids. We will have to consume some from our diet. That's just how the luck of the draw. But I will say the only things you really have to memorize is these 2 general diagrams, and then the specific pathway for this diagram in the pink box, because Solomon went into it much more in depth. Yeah. And then we'll go over this one as well between the glutamate and glutamine. There's a pathway specifically for that, and we'll talk about the regulation on its own separately as well. So in terms of amino acid biosynthesis, we have to use our glutamate-glutamine system. So on this middle diagram right here, we're having the synthesis of glutamine from glutamate. So as you can see here, we have glutamate, it does its thing. You should know this pathway very well. You should have basically every part of it memorized at this point, where we're going to activate our glutamate with the phosphate group. The phosphate is going to get attached to the carboxyl group. It makes sense. It's a carboxyl group. It can get added to a phosphate. Then once we have the phosphate, it's higher in energy, right? Because the phosphate group allows for that. And the ammonia group can attach itself and form that amide. And then in the right diagram, this is now talking about incorporating that ammonia that we just added to our glutamate onto our glutamine, whatever it's called. We're going to pass that ammonium to a different molecule. It could be this as another amino acid, it could be to some other one. So this is exactly how we're going to be able to pass it on to, let's say, an alpha keto acid and build up our amino acids from there. It's going to be this very general scheme. And in terms of mechanism, I mean, it's pretty straightforward. It's just a nucleophilic attack, and the ammonia just gets shoved off of it. But that's pretty much it. You just really realistically have to know that ammonium can get transferred, and this is technically like a deamidation reaction. So does anyone have questions about this slide at all? Okay, but because amino acid synthesis stems from glutamine, it's very highly regulated. And so that's what I want to talk about here is we have a bunch of molecules that are able to negatively impact, to do this feedback inhibition and stop this process from occurring. And this makes sense. If we have a lot of product, we don't want to make more of it. Now, there's this one special one that you guys should definitely be comfortable with doing, and it's the adenylotransferase enzyme that's able to alter glutamine synthetase. So we know adenylotransferase, its main function is to inactivate glutamine synthetase. So you can just say at baseline, glutamine synthetase is active. And then with this enzyme, it's going to inactivate it. But what we know is that uridynotransferase is going to inhibit this adenylotransferase. And when it inhibits that, it's going to stop this arrow from happening, right? So our glutamine synthetase is going to be active. So again, if we have this substrate present, it's going to inhibit this enzyme, and this arrow is no longer going forward. So we have our active state being favored. Now, there's other conditions where we can either promote more of the substrate to occur, or we can inhibit and have less of this substrate. And that's through these varying conditions, right? If we have our alpha-ketoglutarate, if we have a lot of this makes sense. Our alpha-ketoglutarate is our alpha keto acid for the proteins that we want to make. If we have a lot of the substrate and we want to convert it to amino acid, we want to stimulate this. So if we, or sorry, no. Why does it stimulate? Oh yeah, sorry. I was confusing myself. So alpha-ketoglutarate, if we have a lot of the substrate, that means we want to form our amino acids. So we're going to stimulate this transferase enzyme. And what's going to end up happening is because it's stimulated, this is extremely inhibited. So this arrow is, let's just say it's at 0. This is not occurring. So our glutamine synthetase is 100% active. Now, because it's 100% active, we're favoring the amino acid biosynthesis pathway. So I think it'll be better if I say the other direction as well now. So for the glutamine, right? Let's say we have a lot of glutamine. That's our products. If we have a lot of glutamine, it's going to inhibit this enzyme. And what ends up happening if this enzyme is no longer working, there's no inhibition here. So the arrow is now working 100% efficiency to inactivate glutamine synthetase. So the end result, if we have a lot of glutamine, we don't want to make more of it. And so it's going to do through this like inhibitor of inhibitor pathway. I know that's like a lot of words thrown at you, but just follow through. Like if we have a lot of product, it doesn't make sense that we want to make more. And then just walk yourself through it. If we're inhibiting this transferase enzyme, then the transferase enzyme is no longer inhibiting this one. And then that means the arrow is going at 100% that way. And so we're favoring the inactive state at this point. Does anyone have questions? Yeah.
I'm confused how like glutamine is linked to amino acid synthesis because I thought they were just like transferring and it like constantly switches between glutamate and glutamine. So like where is it measuring the high glutamine levels?
It's measuring within the liver. So remember that everything in the liver, I'll go back to this diagram. Everything in the liver ultimately gets converted into this glutamine, glutamate, these two like amino acids, alpha keto acids. So this is the main one that most of the cells use, the liver uses. And so there's like a fine balance, right? If we have a lot of ammonium, right, we're going to favor more glutamate formation because it's the carboxyl group. It's not the glutamine showing here with the amide. Does that make sense? So the reason why it's used for amino acid synthesis is because that's, again, in the reverse direction. If we favor a lot of glutamine, that means we can build more of these amino acids, and it can form these other ones. Again, it doesn't form all 20. It forms some of them, but it's mainly this pathway because most of the cells use that in the first place, the glutamine. So it can go back to the tissues and then build up, respectively, their amino acids and proteins. Any other questions? Okay. So just to give an overview of the amino acid biosynthesis, there's a lot here, and the way they've tested this before in previous years, just to give you a preface, is that they would box some of them, not all of them. They would box some of these boxes, and they'll say like figure out what the amino acid is, and this is just like a straight up memorize the chart, which is so like dumb because this. Yeah, it's so dumb, but they'll literally like just block out these boxes and then say, what's the amino acid for this? Or I think last year they boxed out the 3-phosphoglycerate and you had to determine what the immediate substrate was. So that's how they're going to test you in terms of this diagram, is they'll literally just box them out and you get to like have rote memorization for that. But there is one pathway that you guys will have to know, and it's the serine to glycine cysteine, which we will cover much more in depth. You do have to know the mechanism for that. Yeah, and then the whole point of this diagram is just saying that we're using precursor molecules from glycolysis, Krebs cycle, and the pentose phosphate pathway, which, I mean, that's just kind of self-explanatory. The cell's going to use the metabolites to build things up. It's like gluconeogenesis, it's like fatty acid synthesis. So now focusing on the specific, like, magenta box, we are going to basically have our 3-phosphoglycerate. It's going to become serine, and then the serine can have two options. It can either form glycine or it can form cysteine. We'll go through each one separately. So for this slide, we're going to go from serine to glycine. And this one you guys should know extensively, where we have our 3-phosphoglycerate. It gets converted with addition of a glutamate. And what will end up happening is the 3-phosphoserine when it combines the 3-phosphoglycerate with the glutamate, it's going to ultimately end up forming serine. When serine is formed, it can then proceed to become glycine via the serine hydroxymethyltransferase, which, if you look at the difference between this, you see that serine has this R group, right? It has the methyl and the hydroxy group. And we see that in glycine, it doesn't. So that's exactly why the enzyme is named that, serine hydroxymethyltransferase. And a good tip, if you guys are ever stuck with an enzyme name that you don't know and this seems complicated, just draw your substrate, draw your product, and see what exactly is changing. Because oftentimes, it's going to be named after what's being transferred, especially for amino acid catabolism. As you can see here, we have hydroxymethyl. So again, the hydroxy is the OH, the methyl is obviously the CH2. And that's going to be transferred. So as you can see, from the substrate to product, we just name after the substrate, and then you have your enzyme name. So that's how a lot of these names are derived. And that's just a good rule of thumb if you don't have it off the top of your head. Memorize substrate, memorize the product, and then figure out that conversion between the two. Does anyone have questions about this pathway? I know I kind of went over it really quickly, but you should know that it uses it creates cofactors. There's aminotransferases that incorporates that glutamate and converts into its alpha keto acid. And then ultimately the final fate is that you're going to use a phosphatase to get rid of the phosphate group, and that forms your serine, which it could now just be used as an amino acid, or it can create glycine from there. Okay. And then the third is this alternative route where, let's say, instead of forming the glycine, we want to form our cysteine, right? So realistically, the only things you really need to memorize is that methionine, and I know this is a very complex diagram, but basically we have our methionine, which is basically consumed. So we can't just make methionine on our own. Methionine comes from our diet, and you can just think of it like this pathway essentially, but it just forms our homocysteine, right? When it forms our homocysteine, that could be combined with serine to form this very complex cystathionine, but it's just a complex molecule, you need to know that. And it gets broken down using a lyase to form our cysteine. And again, it uses PLP, and as I mentioned before, PLP is a transaminase cofactor. That's what it's usually used for. Does anyone have questions about this mechanism? You don't really have to know the arrow pushings for this at all. Honestly, you don't even have to know these enzyme names. You just have to know that methionine comes from our diet. It gets converted into homocysteine. Once it gets converted to homocysteine, that combines with serine, and you end up forming your cysteine. Any questions? Okay. And then here's an example of amino acid biosynthesis. You don't have to memorize every part of inhibition in this. The point of this slide is this enzyme multiplicity, right? Enzyme multiplicity, all it is if we focus on this top part of the diagram, there are 3 different isoforms of this enzyme that convert aspartate to aspartyl beta phosphate. So there are 3 different isoforms, and they are inhibited in different ways. So this allows for example, if we have maximum inhibition of all these isoforms, then A2 is going to be the predominant because there's no inhibition. A3 would be the next in terms of highest level of activity, and then A1 would be the least active because there's 2 places of inhibition. So that's all that enzyme multiplicity is that you have different isoforms of the enzyme forming your product, and those different isoforms interact differently with inhibitors. And that allows for these modulating and attenuated responses and activities of substrate converting into product, or vice versa. Does anyone have questions about enzyme multiplicity? That's all that this diagram is just trying to show. You don't have to memorize all the specific things. Yeah.
It says there will always be at least one stream of synthesis. Is that true all the way down, or I'm looking underneath homoserine?
Yeah.
It seems like there's an inhibitor of both of those.
Yeah.
Will it still keep going even if both of those inhibitors are activated, or will it?
Yeah, I think the trick is that methionine, it comes from our diet, right? So if we're not forming any methionine in our body, it won't be able to inhibit. So it's we have that base level of activity, if that makes sense. I don't know if that makes sense or not.
It has to be going through, otherwise it
Yeah.
won't be inhibiting itself.
Yeah, exactly. But yeah, it's never going to turn off completely. There'll always be some baseline function, essentially. Does that make sense? Okay. So moving on to the nitrogen cycle, we'll go quickly through this because it's not tested that much and it should be pretty quick. So there are 3 stages of this. There's nitrification, denitrification, and nitrogen fixation. This diagram doesn't do it that good of a job. It's this one that I really like the most. And I'll start from the top of the diagram where we have nitrogen from the atmosphere. So the first thing that happens is, much like carbon fixation, right, from the atmosphere, we have nitrogen fixation. So nitrogen fixation, all it is that we're converting this atmospheric nitrogen into ammonia. And there's bacteria that does this. You don't even need to know the specifics. Just know that nitrogen fixation just converts it from an unusable natural source to a usable, viable source now. And so when it converts into ammonia, again, we can either build up amino acids or we can undergo nitrification. Nitrification, all it is converting this ammonia, which has an oxidation of -3, to an oxidation state of +3. It's going to oxidize your ammonia into nitrite. And what can end up happening is with nitrite, it can form nitrate. I don't necessarily know what the significance of it is, but just know that that's what the process is, nitrification, where, again, you have a nitrogen that has a lot of hydrogens attached to it. So that means the nitrogens own all the electrons. And then now you're adding the oxygens, which we know oxygen is much more electronegative than nitrogen. So it basically the oxygen will pull the electrons from the nitrogen, and therefore the nitrogen doesn't own any, or doesn't own that much. And then the last step is denitrification, where we convert this nitrite, it's oxidized, back into atmospheric nitrogen. And that's just, again, through denitrifying bacteria. You don't need to know that. You just need to know specifically what these processes do. So nitrogen fixation, as we went over, it's atmospheric to ammonia. Nitrification is ammonia to nitrite. And then the last process, which is denitrification. Does anyone have questions about this? That's realistically what you have to know about this. Yeah, this slide's just more confusing. Okay. There are 2 nitrogen fixation enzymes. So the first one is dinitrogen reductase, and then the second one is dinitrogenase. I mean, you can know the specifics, but honestly, like, the only things you really have to know is that it requires 2 ATPs per electron that's formed, and then denitrogenase is essentially just converting your atmospheric nitrogen to the ammonia, given the electrons that it has built up on it. So it's just in tandem how it works together. Does anyone have a question about that? This is like not going to be tested probably. I'm just covering it very briefly. Okay. This is another slide just showing the electron flow between the dinitrogenase reductase and the dinitrogenase. That's basically all it's showing is these electrons moving through. You end up using ATP along with this to reduce the dinitrogenase, and that ultimately gets you to converting the N2 into 2NH4+. And honestly, I don't think you would be testing on like numbers specifically like 8 and 16, but I mean, they're multiples of each other. The only thing I will say is perhaps you may have to know the 6 electrons, if I'm not mistaken, which is the nitrogen fixation. Like this equation, probably the only thing you realistically have to know, which is N2 plus 6 electrons plus 6 protons. It's going to convert into 2 ammonia molecules. But that's just like stoichiometry and like balancing stuff out. But that's realistically the only equation you would have to know. And this correlates with that, right? As you can see, the 6 electrons on the bottom right here, where we convert that nitrogen, atmospheric nitrogen, to ammonia. Okay, and then these are practice questions for slide 15, and yeah, these are just a bunch of practice questions that you guys can go through. Straight gonna get you.
Sounds way. Yeah. Any questions on that lecture at all, or 13 to 14?
Or like, you guys want me to go over another thing again? I can as well. Like, Walsh Ray is coming back because he's gonna do lecture 16.
There he is.
Oh, never mind. There he is.
No, you're good. Very dramatic.
It's like another part.
No, you're good.
Alrighty, hi everyone. We're going to go over lecture 16, integration and regulation of metabolism, which I think is a really important lecture in terms of the physiological effects of everything that we're talking about. We know our professors like to talk about these like physiological states, what contexts will your cells be in terms of like being fed, unfed, things like that, and that's what we want to prioritize with this lecture. I'll try to go through it a little quickly because I know it's almost 9. Yeah, okay, so overview. These diagrams are not necessarily things that you're going to have to recount on the exam, but they combine a lot of the processes that we've learned so far into one, and sort of just indicate where ATP is used, where ATP is made, and that those patterns are going to reappear. in this unit as well. Okay, so really quickly before we get into specifics, I want you to know 3 major ways that we regulate metabolism and be able to explain them. Number 1 is pathway regulation. We've seen this time and time again, how different products affect or regulate the enzymes that actually create them. So be able to explain that cellular theme. Number 2, subcellular compartmentalization. We might have not said this explicitly in the class, but we've seen it time and time again with the cytoplasm and the mitochondria. And there's so far one example that we've learned where that compartmentalization is kind of blurred, and that's in the urea cycle, where you have the cytoplasm and the mitochondria. So be able to describe examples of that. And then finally, organ division of labor, which is the emphasis of lecture 16. And so you'll need to know preferences of certain organs, what they're able to interpret, what they're able to export, and there's going to be a really good chart for that later in the slides. This is the key diagram about organ specialization. You're not required to know what every single diagram in here is indicating, but be able to have maybe 1 or 2 examples ready to use, just so that professors know that you know the overall organ level implications of this content. Okay, let's move into specifics and talk about the pancreas first. Pancreas, we know, secretes hormones in response to blood glucose concentration. For meals, when you eat meals, your insulin is going to be high. Try to bring back that glucose into your cells while also stimulating some of that glycogen formation. And then fasting, the opposite happens. Your blood sugar is really low, and so glucagon is going to be high. And so when you create your mental flow charts or even your physical flow charts, try to test yourself with conditions like low or high blood sugar, starvation, being well fed, survival, and talk those pathways out, whether with a friend or by yourself. But those overall steps are going to be asked in discrete units. And so it's important to think of them holistically as well, not just one by one. In terms of our adipocytes, there are white and brown. And so they store and supply fatty acids mainly. But our brown adipocytes actually have another function, which is thermoregulation. We saw that in exam 2's content in terms of uncoupling and heat production. That's a really important cell type where that happens. Moving on to the brain. Think of the brain as a leech. The brain is very specific, has very rigid preferences on what type of energy forms it likes to have. And that's what makes it such a fragile organ as well. The liver, on the other hand, is a lot more flexible, and it does a lot of things as well. It processes sugars, amino acids, lipids on the daily, whereas the brain prefers just mainly glucose, and when it really needs energy, it might move on to ketone bodies and fatty acids. And the liver, obviously, we know does gluconeogenesis as well. And so if I was able to advise you on how to organize these things, think about preferences and which one is the least, moving from least flexible to most flexible is a good way to think about these different organs. So starting off with the liver, sugar metabolism is just review for us. We've seen different fates of glucose. Be able to recount these. I guess this is a component of the class that's cumulative and shows up again and again. And so be able to know examples of export, storage, energy production. Again, all familiar, but good things to have readily for a short answer response. Amino acid metabolism, I won't spend too much time on this because we talked about it quite a bit. But again, just to emphasize, know where your proteins are processed, know how they're transported. We saw the whole glutamate fiasco. We saw how amino acids also are precursors to a lot of different biomolecules. All review, but the liver is the focus here. The liver is the one processing these things. We also got lipid metabolism we talked about very early on. Again, not too much emphasis on the diagrams that we're posting on these slides, but being able to know where the processes exist, like the cytoplasm, the mitochondria, and what's being exported, what's being brought in. Those are the key things to know. I would highly suggest making a chart for that or being able to create a checklist for those things. Okay, the brain, which we haven't talked about too much until this lecture. The brain prefers glucose as its main energy source. However, during starvation, when glucose is unavailable, the brain will use ketone bodies. But this is not generally a good thing, and it is very unsustainable. metabolizing ketone bodies creates a really heavy like acid load, and that is toxic to our tissues and our cells. You can also imagine the brain requires a large amount of ATP, and the reason for that is because we need to be able to power that sodium-potassium pump 24/7. We know our brains and our neurons sort of thrive on having this negative resting potential, and so if we can't maintain that over time, then we won't be able to have any cellular function, really, or bodily function. Alrighty, muscles. So muscles are a little bit more flexible. So most of the energy sources, as we know, are going to be used for contraction. During light activity and rest, it can use a wide variety of things. Fatty acids, glucose, ketone bodies, not necessarily like a huge difference in what it prefers, but if you wanted to create a checklist or a pathway, it could be like glucose a little bit more readily available, maybe then tapping into some of your glycogen storages that are in your muscle, and then maybe fatty acids and then ketone bodies, if I was able to give you like a loose preferential hierarchy. So that's during light activity and rest, but when you're in heavy activity, you need energy very readily, and you'll be able to cause that contraction quickly. And so you're going to use a molecule called phosphocreatine, which is sort of stored ATP. It's a reservoir of high-energy phosphates. It's created from creatine plus ATP, and when that phosphocreatine molecule is hydrolyzed, you're able to get energy and contraction really quickly. And so that's just an example of what might be used for heavy activity. You can also imagine that might be during like anaerobic exercise as well. Okay, the Cori cycle is probably the most important part of this whole muscle-liver crosstalk. And so this cycle allows your muscle to maintain proper levels of glucose. Again, we can imagine that during heavy exercise, you need your muscles to have a proper level of glucose, and you don't want a lot of these waste products or byproducts to accumulate. And so luckily, we have evolved to find some way to allow those byproducts to reach the liver and go right back up to the muscles. The way that happens is through lactate, which wasn't always in the best light, but now we figure out a way to shuttle the lactate quickly into the liver, get converted to glucose as a precursor to gluconeogenesis, and that glucose will be shuttled back into the muscle to make sure those glycogen storages are never depleted. This cycle is very convenient. It's very useful. It's payoff, and remember, this is a trope in biology. Fast cycles have costs. So they have cons as well, and the con in this case is that it uses 4 ATP per cycle. And so while it's fast and convenient, it helps you sustain energy and fuel sources during heavy exercise, it also costs 4 ATP to make this whole thing happen. And a lot of that ATP usage comes from creating glucose from your lactate, which is a process that we've seen before. And after heavy exertion, so now we're moving away from an anaerobic environment to an aerobic environment, you start to increase your O2 consumption, and you can replenish your phosphocreatine for when you need it, and you can also keep your muscle glycogen at a generally like homeostatic level instead of this continuous cycle of replenishing. Okay, this is the Holy Grail diagram in terms of fuels of these major organs. You should know the preferred fuel. For example, the brain is glucose, might use ketone bodies when it really, really needs energy. The heart muscle, interestingly, prefers fatty acids. We don't have to worry too much about why. And then also recognize what organs can export things. The liver can do this quite effectively, but organs like the brain and the muscle and the heart muscle might not do this so much, with the exception of the lactate Cori cycle that we just learned about. Okay, so this is how I think we all should be thinking about metabolism, and I think we should be using the two extremes. So we have the fed state, the fasting state, and those are the easiest way to see these effects. The fed state, we might have our dietary fats, which can be sent directly to the muscle and heart for energy, as we just saw in the diagram before. But the fats can also be sent to the liver to be metabolized, which we learned about in lectures 13 and 14. These can be turned into your storage lipids and then stored for later use. We also know that our insulin levels rise via the pancreas, and that helps us with a little bit of that glucose shuttling into your cells. The liver will uptake that glucose, and some of it will be used for storage. Some of it might be sent to the brain for ATP production. It depends on what the cell needs or the body needs at that time. And so I heavily encourage you guys to not use absolutes when you describe these topics. because it's almost false to say things like always or not, because it depends on the cellular context. And then proteins, like we learned in lecture 15, when we take out certain groups from them, they can be used for biosynthesis pathways, but also important to excrete them effectively as well. For fasting, which is the other extreme, you have glucagon being secreted from your pancreas, and that will help us stimulate gluconeogenesis, but also help us break down glycogen into glucose. So those are the two ways that we know how to replenish or keep our glucose levels at a homeostatic level. In addition to this, our triglycerides are released from the adipose tissue and sent to the liver to produce ketone bodies, which is that last resort fuel source that our body can use. Proteins in the muscle are broken down. This is an indication that you have no food intake for a pretty long time. And we know that some of those proteins, when they're broken down, can be converted into things like pyruvate, oxaloacetate, and therefore can be used for gluconeogenesis and ketogenesis. In this case, I think a key idea here is that you actually slow down the TCA. And this makes sense because in a fasting state, why would you want to prioritize something that is breaking down things for energy? Or why would you want to focus on creating intermediates that you might not necessarily need to use at that time? You want to focus on your fight or flight response, which is creating that energy as quick as possible. And so therefore, things like TCA might slow down, but those resources that you do have might be used for things like glycolysis to keep energy levels high, and other intermediates might be used for gluconeogenesis, like pyruvate and oxaloacetate. And so there is a balance between biosynthesis and breakdown of compounds. And in the fed state and the fasting state, you have to think about which of those processes are you leaning towards. And that will come up in terms of explanations. This is just another diagram to show exactly what we were talking about on the last slide. Pink rectangles, you should know where these things come from. And that's about it for this. Okay, let's talk about hormonal signaling, which is where a lot of these responses come from. This allows us to regulate a lot of metabolic pathways. And there's two major ways that we've learned so far about signaling. One is cell surface receptors, and the other is nuclear receptors. Know which types of molecules prefer these types of pathways. You can imagine a cell surface receptor must interact with a molecule that can't get through, so something that's big, charged, and a peptide hormone, but something that's nuclear, or something that's able to interact with nuclear receptors, which are tucked away inside the cell, must be able to get through the membrane, something like cholesterols. And so know which categories that fits into. Cell surface receptors have extracellular binding sites, allowing them to interact with these hydrophilic, big peptide molecules. And they can be broken down again into ionotropic and metabotropic. Ionotropic is a little bit more simpler, that when you have ion flux, your cell is able to interpret those changes and cause some type of response. Metabotropic is a little bit more nuanced, has a little bit more variety in terms of second messengers. There are so many second messengers. I think one example you should always have on you at all times is cyclic AMP. In terms of nuclear receptors, you're now going to be binding to intracellular receptors, and then that's going to cause some direct change to transcription or gene expression. Usually, a lot of these pathways are going to cause altered transcription of specific genes, whether or not they're cell surface or nuclear. There is not too many exact effects that we need to worry about. We learned a couple in the whole insulin pathway, which we'll get to in the next couple of slides. Glucagon, insulin, I discussed most of this, so I'm just going to leave these slides here. I would recommend not losing sight of the enzymes that we learned about in exam 2, because they are the ones that are allowing us to be pro, glucose forming and pro glucose breaking. And so don't lose sight of those enzymes, because they might ask about those processes through those enzymes. For example, PFK being prioritized, or the gluconeogenesis enzymes being prioritized. Just another representation of the same content with a little note about epinephrine, which is secreted by the adrenal medulla in response to low blood sugar. So it's a similar effect that glucagon has. But I guess you can say also that indirectly it also regulates insulin by suppressing production of insulin. And so it has the overall same net effect as if glucagon was affected, which is why in a lot of the diagrams in the last exam, we saw that green arrow for both epinephrine and glucagon. Yeah, this is pretty standard. Just know epinephrine is going to stimulate glucagon secretion and inhibit insulin secretion. Okay, diabetes. So for diabetes, know the two major types. Type 1 is autoimmune, which causes destruction of your insulin-producing cells, which are your beta cells in your pancreas. And therefore, you can't synthesize insulin. And so this is why this is called insulin-dependent. Whereas type 2 is a little bit different. You can still form insulin, but the cells are not interpreting it properly, which is why it's called non-insulin dependent, or another phrasing you can use is insulin resistant, indicating that you still form it, but you're just not sensitive to it. One of them happens early on in life. Other is necessarily develops later on in adulthood. And so there are some effects that diabetes causes that is detrimental to the body. And so number one is elevated blood sugar. You can imagine this is going to be one of the immediate things, because if we're unable to create insulin, which we know causes transporters to be secreted to the plasma membranes of your cells, if that's not there, then you have no transfer mechanism into your cell. And that's for both not having insulin and not being able to interpret that insulin. And because of that, the issue is that your cells don't really know that they're diabetic, right? They just understand that there's no glucose in the cell. And so because of that, you have overstimulation of gluconeogenesis, which is an issue because there actually is a lot of glucose in your blood. It's just not being pushed into the cells where you can start to use it. So that's the second big impact. Clinically, that leads to things like hyperglycemia, glucosuria, dehydration, and osmotic diuresis. Long story short reasoning is that when you have a lot of solutes, in this case sugar, in your blood, you're going to want to excrete it so that you can maintain those solute levels. But in excreting a solute like glucose, you require a lot of water, and therefore that leads to dehydration. And so that connects those clinical symptoms. And then lastly, again, in addition to the body thinking that it has no sugar, which is a false understanding, it's going to think that there's no fuel source right now, and it's going to produce ketone bodies. And we know ketone bodies, again, are like a survival instinct. They're very last resort, and that's going to lead to ketoacidosis, because now your body's going to rely on ketone bodies to generate ATP and to maintain cellular function. But ketone metabolism, it creates acid load, and that leads to diabetic ketoacidosis, which is not pleasant at all. So those are the three major effects of diabetes if it's not controlled, and the way to control that is by providing insulin. So, yeah. Okay, so now I'll talk about signal transduction a little deeper. Signal transduction is important for a few reasons, and you should be able to describe these overall biological tropes in a couple sentences or phrases, and those tropes are specificity, amplification, adaptation, and integration. So those are four major qualities or characteristics of signaling molecules and receptors. And the goal overall is to amplify that signal and convert it to a cellular response. And for the sake of our class right now, cellular response is going to be some type of gene expression alteration. Don't worry too much about this diagram to the left. We're going to go into depth about some of these receptors now. First one is a GPCR. In this case, epinephrine commonly uses GPCRs. Okay, I'll stay on this slide for now. So there are a lot of steps in GPCR. If you have time, you should try to go through each of these 7 steps and be able to generally describe them. But if you're able to create a shortcut summary for yourself, first think about what happens before a signaling molecule binds to GPCR. Key is that they're bound. The different G subunits are bound to each other. When a hormone binds to the GPCR extracellularly, there's a dissociation of one of the more active subunits. The active subunit is the alpha subunit. Alpha subunit is special in that it's not just a component of the GPCR, but it also has enzymatic activity. And that enzymatic activity is associated with a GTP molecule. And when that GTP molecule is bound to the alpha subunit, it can cause activity and allow that signaling pathway to happen. And then when you've accomplished what you wanted, that same alpha subunit is going to regulate itself and hydrolyze that GTP. When that GTP is converted to GDP, it starts to shut down the pathway and then bring your two subunits back together and allow for the next round of signaling when it needs to happen. And so this idea of self-regulation that the alpha subunit has is very important to be able to explain because it's like this two-pronged benefit. It's able to recognize when the pathway needs to stop and then halt it on its own. Cool. A further example of GPCR is cholera. And so what cholera actually does, it locks the alpha subunit in its active state, which is not good because we know that constitutively active pathways are also not helpful to the cell because that's using up a lot of energy and a lot of metabolic power. So this is an important example of being able to recognize what happens when you affect something in this pathway. And so you could be easily asked about, hey, what if I took away the hydrolytic activity of the alpha subunit? What would happen to my pathway? What if I kept providing it GTP? What would happen? What if I blocked the receptor of epinephrine? What would happen? So things like that, be able to connect them to their downstream effects and think about what would accumulate and then what would happen to the cell in the downstream. Okay, epinephrine is a GPCR mechanism. Insulin is actually a receptor tyrosine kinase mechanism. And as the name suggests, this receptor is active because you have a phosphorylated tyrosine residue once you get binding with that receptor. There's a lot of jargon in this diagram, but in terms of connecting it to the terms that we should know about receptors, in terms of amplification, the key here is that one receptor, tyrosine kinase, can actually activate multiple IRS molecules. And you can think of your IRS molecules as these further downstream messengers. And because there's so many of them, or there's different types of them, we can cause different cellular responses. And the reason why I mentioned that you can have multiple cellular responses is because the insulin pathway is actually not just important for glucose transport inside the cell, but it's also important for cell division and DNA replication. And so now we've learned two ways that insulin impacts the cell, and it's not just always in glucose, but it's also for allowing your cells to replicate appropriately. And so be able to have those two ideas ready to go. Okay, insulin also affects glycogen synthase, which is one of the things that we know about in the fed state of a cellular environment, that not only do you get glucose shuttled into the cell, but what you do with it is you store it for later use. The key with this pathway is that it actually involves a lipid in the bilayer itself, which I think is really cool. So far, all the pathways that we've learned about include messengers that are found either attached to that inner membrane of the plasma bilayer, or it's a cytoplasmic second messenger. But in this case, it's actually a lipid within the bilayer that has a signaling role, and it's PIP2 being converted to PIP3. And so it's the role of this molecule that's able to activate, one of them activates and the other one is able to inactivate this whole process. If you have time, try to go through the 5 steps of this, but here recognize that just insulin has multiple effects on the cell. We've learned 3 now: glucose intake, cell replication, and then also glycogen synthase. Talk a little bit about gated ion channels. We've probably learned about this in all of our bio classes, but it allows ions to move down their concentration gradient. Voltage-gated channels are the ones that we find in neurons. They allow us to create enough potential to release neurotransmitters. Really briefly, it happens through depolarization of the membrane. Keep in mind that the membrane is originally negative, and that's because of that sodium-potassium pump. This depolarization will cause voltage-gated calcium channels to open. Calcium increase in the cytoplasm is usually a sign of activation, which is going to cause neurotransmitters to be released and cause other effects on nearby cells. Nuclear receptors, as I mentioned before, are activated by steroid hormones because they're small and they're able to pass through that membrane easily. But they usually do need to be carried by some binding protein in the bloodstream, and we can think That's important because for a small hydrophobic molecule, it could be lost pretty easily. So just in our themes about like activation and compartmentalization, it's important to bind it or tag it so that it knows that it needs to reach a nuclear membrane. These are going to directly regulate gene expression, not have to go through a secondary messenger system. And specifically, I think one buzzword that is important that is going to bind to the hormone response element within the nucleus and lead to direct expression change. We also talk about really quickly receptor guanylyl cyclase. This is one of the receptors that causes a conversion of GTP to cGMP, which is just one example of another second messenger. We learned about adenyl cyclase and some of those GPCR pathways, which converts AMP to cyclic AMP, and that causes transmission of a signal towards the nucleus. This is just one more example using a different secondary messenger molecule. Alrighty, so just to end off with lecture 16, application is very important here. Our professors have this like mindset of being able to recognize what happens when you make a change to a system, and I think this is where the content is the most important. Think about when you take away food or diet. Think about when you have too much food. Think about what would happen if you took away a certain hormone, if you took away its receptor. These extremes are a really great way to study and then see how the body would respond. Be able to describe the signal transduction pathways, especially those 4 main qualities or characteristics that we care about. And then finally, that one diagram that had the organ profiles about what fuel sources they prefer, which ones they can make, and which ones they can export. Very key. You can be asked about that through various ways. And then there are some practice problems, but we will go on to lecture 17.
Okay, this is going to be one of the more complicated lectures. So there's a lot of pathways in this lecture. I purposely went through all the pathways and just said, like, this is the things you need to know, and I highlighted them in magenta boxes. So we'll go through everything very slowly. So before we go into the nucleotides themselves, so there are going to be 2 groups that we talk about, 2 families. There's going to be amines, and there's going to be tetrapyrroles. But we'll go through each one separately. So first thing we'll talk about is amines. So this is just a practice question. We won't go through it right now, but these are different types of biological amines, and they may just ask you, which ones are they specifically? And, I think at least my year, but my year was like a bad year anyways, but they had us like identify these specific ones like dopamine. I'm pretty sure this is norepinephrine on the middle left, and then epinephrine's on the bottom left, and so on and so forth. But the main characteristic of this is that the amines, they don't have a carboxyl group. They've been decarboxylated, and that's going to be the key for making these amine molecules in the first place, is that there's going to be an enzyme called a decarboxylase that gets rid of that the carboxylate group, and it's going to form this amine where it's just positively charged, and it's the only thing on this carbon. So there's a lot of things here. The only thing you have to really memorize is this red box. The rest of it I'm just going to talk about and hint at. And it seems like a lot, but this is pretty intuitive, I want to say, with tyrosine converting into dopamine. It's not necessarily you have to memorize the structures. It's just that tyrosine, again, has the group here, and then we know that it's decarboxylated from it. So it's going to be some form of derivative of it. And the point that I'm trying to get at is that tyrosine can become dopamine, it can become norepinephrine and epinephrine, because we know that dopamine can be converted to the other 2 downstream. It's like a separate pathway. But the point that I want to emphasize here is that we're just using a decarboxylase enzyme that has a cofactor with PLP. So for all of these, to form an amine, we have to use a decarboxylase with the cofactor PLP. So they may say, like, what's the general function of PLP? We know it's a transamination part as well. We know it does that, but we also know now that it does a decarboxylate to form an amine. So it has either or. Does you have questions about this? It's mainly, the biggest thing is just memorizing your precursor and then what it breaks down into, the possible products itself. Okay. Oh, your question?
Do you have to memorize the enzyme specifically?
No, I mean, they're named after their precursor anyways, so like it's just histidine decarboxylase for this to form histamine. Yeah. Okay, so this is just, don't worry about the mechanism. You will not need to memorize the mechanism. I'm just putting it there for your guys' reference. I just wanted to show in this slide that PLP, this molecule right here, you may be asked to say, hey, like what, they'll give you, you'll have to recognize it essentially on the exam. So they may put a bunch of like cofactors and like the structures of them, and you may have to memorize it. So it's very good to, again, put this on like a Quizlet, just blank out the name and then guess the name, recognize it. That's one of the bigger things. And then another thing I wanted to mention is that, again, it does that transamination reaction, but it also does the decarboxylation. And with amines, we're talking about decarboxylation. You don't really need to know the mechanism about shift base, any of that. It's not really, you're not going to be tested on it. So now moving on to polyamines. Polyamines, all it is that we're adding a bunch of amine groups into a single molecule. That's realistically what it is. So I wanted to emphasize that POP is going to be the important cofactor, but the most important thing is that we're going to be combining just two amines together, and then once it's combined, then we just add other things. So essentially, our first step for any polyamine is going to be a decarboxylase. Once we do that decarboxylase reaction, then we can just use a aminotransferase to combine these polyamines together. So that's what I'm trying to get at, is that in order to start this process, we do decarboxylase, and then to add them like Legos, then we just use the transferase. So it's two different classes of enzymes that form these polyamines. It's just an extra enzyme that's introduced to it. In terms of this, like realistically, the only things you have to really memorize are just the products. And then as I mentioned, there's these two different classes of enzymes. The one that's to start the polyamine formation, which is the decarboxylase, and then those transferase molecules that form these. No, you don't have to memorize like propylaminotransferase 1. You just have to know that there's these two different classes that work in tandem. Is there any questions about polyamines? Okay. So this is one example that you guys will have to know in depth, is the ornithine decarboxylase. So here we know ornithine decarboxylase. It converts ornithine to putrescine, which it's essentially just getting rid of the decarboxylate group. Now, the reason why I bring it up is because we talk about the suicide inhibitor in class, DFMO. And what happens is, I know Solomon shows you guys the mechanism. You don't need to know it, and I'm sure he's mentioned, like, don't worry about the mechanism. All that the suicide inhibitor does, a little review from exam 1, is it forms covalent bonds with the enzyme, and that basically knocks it out. So they may ask you a question of like, okay, how does TFMO act as a suicide inhibitor? It's almost like a callback to exam 1 material. And all you have to say is that it forms a covalent bond with ornithine decarboxylase, and it locks it in place. And now a question I will propose, because this was asked last year, is what happens, or how does the body like circumvent this issue of the suicide inhibitor? And the answer is that there's enzyme turnover, meaning that the enzyme that's inhibited permanently, that gets broken down, degraded. We form new enzyme in its place, and that new enzyme doesn't have the suicide inhibitor. That's the answer to that. And it was asked last year, so I'm just bringing this up now, like now that I remember a little bit from that. But does anyone have questions about polyamines before I move on to tetrapyrols? Okay. So now we're talking about the tetrapyrrole family. There's a lot of pathways. Again, you do not have to memorize everything. You don't have to memorize every enzyme. That is a waste of time. I did that, and it was not tested at all. I really wish I did not. So the common precursor for tetrapyroles is this delta amino livolinate. I don't even know how to pronounce it. But essentially, there's two different routes in animals and in plants to form this precursor, but they converge. So in animal cells, we're going to be combining a succinyl-CoA and a glycine together, and that's going to end up forming that precursor molecule. Whereas in plants, they're able to do it with glutamate, and that's just because of this glutamyl tRNA synthetase. You don't even need to know that. Just know that there's two different pathways for animal cells and plant cells, and they require different substrates for each accordingly one. And another big thing is that the heme is able to inhibit animal cells. So like it's able to inhibit this formation of the precursor. And again, tetrapyroles, they form the heme. That's the end goal. The tetrapyroles form the heme. So if we have the heme, we want to inhibit that initial step that causes the formation of it because of product inhibition. So that's what this inhibition here is. And then this diagram is in terms of like the precursor converting into the heme itself. There's this pathway. You don't have to memorize all of it. Don't make that same mistake like I did. It's a waste of time. You're going to basically go through this multi-step thing, and you're going to end up forming your heme from the delta amino livolinate. And you need 8 of those. That's pretty much it that you need to really know from this slide. Does anyone have questions about the synthesis of this? It's not supposed to be in depth. It's just supposed to be like a bigger picture thing. Okay. So now moving on to, let's say we have the hemes, right? How can we degrade them if we want to? And so when we degrade the hemes, we end up generating either bile or pigments. They're usually combined. So the first thing that ends up happening is the heme, and these are enzymes that you should have memorized, is heme oxygenase will basically decarboxylate this heme, and you'll end up forming your biliverdin. Now the biliverdin, I don't know if you guys know like a little bit of Spanish, but verde means green, and it's almost sounding the same here. So it just means it's like a green color to it, biliverdin. And then it undergoes another reaction with biliverdin reductase, and that forms bilirubin. Now bilirubin, there's not like a mnemonic or something I can give, but it's yellow. And when we have excess bilirubin, that's exactly what forms jaundice, this yellowish color of like the skin. So the clinical significance of bilirubin is that when there's a lot of it, you end up forming yellow skin. And then biliverdin is, I think, green. So when it talks about bile and pigments, the bile itself is just color. That's pretty much it. There's any other questions about the degradation? It's not really like, you basically stop here at the bilirubin. You don't need to know this bottom stuff at all. Okay. So now moving on to nucleotides. This is the bulk of it. I will say, know the differences in nomenclature. It is hyperspecific. Like as in, if you write, for example, you write adenylate versus adenosine, right? And even though you're trying to identify the right molecule, if let's say the answer was adenylate, but you wrote adenosine, we won't give you points, no matter how much you argue for it. You should know exactly how to write it. Another alternative name, which we'll go into in depth, is that adenylate is the nucleotide, so it's AMP or ADP or ATP. You could just be safe writing that. Whereas adenosine, we know it's just the sugar and then the base attached to it. But that is something to keep in mind with this exam when it comes to nucleotide naming. Okay, so moving on to the basics, they probably will, like I can be 95% sure, they'll put a random base on a random sugar. It could either be a ribose or deoxyribose sugar. And they'll put one of the 4 or 5 bases, whatever, on top attached to it, and then they'll put a phosphate group or they'll leave the phosphate group out. And you have to determine, just by recognizing, not drawing it, by recognizing whether it's an adenine, guanine, cytosine, thymine, uracil for the base, and you have to determine whether it's a ribose or deoxyribose for the sugar, and you have to determine whether it's a nucleoside or a nucleotide. Nucleotide has the phosphate group with it, nucleoside does not. So that's like the way you can methodically break these types of questions down in terms of recognition, is first the base, then the sugar, then if there's a phosphate group. Yeah, and then we'll go over all the difference between deoxyribose and ribose in a little bit. But yeah. Yeah, so I'm going to get into that a little bit, but essentially there's 2 types of sugars. There's ribose and deoxyribose, DNA versus RNA. For the deoxyribose, we lack the 2' here. So this sugar that's shown on the screen on the bottom right, this is a ribose because it has that OH on the 2'. Deoxyribose means we lack the 2 OH, so there would be an H here instead. Yeah. So now we'll go into the nucleotide biosynthesis as well. Do you have a question? Oh, no. Yeah. So we'll go on to the nucleotide biosynthesis pathways and all that. You will not have to memorize every single step. You just have to memorize basically like first step, inhibition stuff to the first step, and I'll explain that. I'll make it abundantly clear. So in order to generate our nucleotides in the first place, we have to generate the base, the sugar itself. And so that's what this PRPP is. So essentially, we form PRPP, and then PRPP will diverge. There will be some divergence where you form either the purines or the pyrimidines, and they form differently. So the purines are step-by-step directly onto the PRPP. So it's almost like sticking on Lego bricks at a time, and you're forming this huge thing. It's one by one. Whereas for the pyrimidines you form the thing first, the ring, and then you attach it to the PRPP. One does it stepwise, the other one attaches the finished product. Is essentially what it is. And again, PRPP is like the foundation for this. And we'll go over how to make PRPP, like the synthesis of that in a little bit. I have a picture showing that. But before that, I wanted to talk more so about this divergence right here, where we do have this PRPP, and we can either go to purine or pyrimidine. And I first want to start off with purines, right? So as I mentioned before, we're adding to the PRPP directly. So it's building up stepwise. And what it means specifically is, we'll start with IMP. IMP has the base here, right? It's already formed. And it has the PRPP right here, which is not showing the full structure, but again, PRPP is just the sugar and this like pyrophosphate, phosphate stuff attached to it. So it's this stuff here that's not necessarily like shown clearly. But the IMP has the full base built, and we'll go into the stepwise function. I just want to focus more so on how we can distinguish between AMP versus GMP, because these are two different bases. So these are two different pathways, and I like to think of them as complementary, where if you have a lot of A, you also want to develop a lot of G, and so you'll inhibit A and promote G. I'll explain that right now. So let's say we want to make AMP, right? So we'll go this top pathway. You don't need to know these enzymes. You don't need to know that fumarate leaves or aspartate enters. All you realistically need to know is that it's going to take one pathway to form AMP. AMP is going to inhibit that pathway through product inhibition, and this makes sense. You don't want to make more of it if you already have a lot. And then the thing about this is that it's going to use the alternative nucleotide to drive this reaction forward. Because again, it's a synthesis reaction. Synthesis, we need energy. So if we're trying to make AMP, it doesn't make sense that we're using ATP, because we want to build that thing up in the first place. So we're going to use the complementary, which is GTP, to power it. So in the same thing in the bottom, for example, and honestly, like to get like full points, I'm pretty sure like you can argue that you can just say first step inhibited by AMP, promoted by GTP. First step of AMP synthesis. Like they're not going to take points off for you not knowing the name fully. And then now moving on to the bottom, we see here that we have IMP, and it's going to get converted into GMP. That's the end result. And so what ends up happening here is that we're using, instead of the nucleotide powering in the same step as the inhibition in the first step, for GMP, it's in the second. But again, the same thing I want to highlight is that we have GMP here. We're going to use the opposite, the complementary nucleotide, to drive the synthesis reaction forward. So instead of using GTP here, we use ATP. And then for right here, the GMP, it's going to do feedback inhibition, product inhibition, to stop the IMP dehydrogenase from favoring this pathway. So it's like it works in tandem, right? It's like a gradient. Like it'll favor one pathway or it'll favor the other based on product inhibition, based on the levels of the pools of AMP or GMP. Does anyone have questions about like the synthesis or why it picks a specific direction? You don't need to know the enzymes like adenylyl succinate, synthetase, or lyase. You don't need to know that stuff. You just need to know where it's regulated, and that's the first step, and that it gets powered. That's pretty much it. Okay. So now talking about how we go from PRPP, this, to the IMP. That's what this slide is showing. You do not need to know everything, and what I'm showing with these pink boxes is that there are 5 distinct carbon sources, and I believe that you guys should definitely have this part memorized for sure, where they may ask you, hey, fill in the thing, where is this carbon coming from? Which substrate? And you have to say, oh, it's forming. Like they'll have it just blanked out. So like definitely have this stuff memorized in terms of where the carbons or nitrogens come from. So that's what this is showing right here. No, you don't have to know the mechanism. I'm just showing like how they add into it in the first place. But realistically, if you know this, that's all you realistically need. So in this, like I'm just showing that glutamine adds, that's one unique thing. Glycine adds, that's a second unique substrate. The formyl H4 folate adds, that's a third one. Glutamine adds again, but that's not a unique one. That's just another one. So it's still 3 unique. Then we add aspartate. That's the 4th one. And then the last one is, if I can find it, I believe it is another formyl. No, it's not. Let me see. Damn, 5. Is it 5 unique? Or maybe I put a typo in there. I think it's 4. Oh, it's a carbon dioxide. That's what it is. I just didn't highlight it. I'll go back in the slides, but free carbon dioxide is the 5th carbon source that comes in. But yeah, no, in terms of like the enzymes, you don't need to know every single one. That's not worth your time at all. Just know that, again, remember this part and then you'll be fine. So now talking about purine synthesis. Oh, question?
It says that it comes in as bicarbonate. I know they kind of go back and forth, but do we have to be specific by saying it comes in as bicarbonate?
No, I don't, you can say either or. I mean, we get the idea.
Yeah.
Any other questions? Okay. So in terms of regulating purine synthesis, so we have to do this at the committed step, and we haven't gone over this yet. So the first committed step is this complex thing, right? And no, the point is not to memorize the complex thing. All it is the first step of PRPP converting into 5-phosphorylribosylamine. You don't even have to memorize that. You just have to memorize the first step that PRPP undergoes is the heavily regulated step. That is it. So IMG, GMP, AMP all inhibit that committed step, and this makes sense. when we're going the stepwise fashion, if we have a lot of AMP or GMP or IMP, that means we have a lot of product and we want to stop the product from being formed because we already have too much. But again, this is a memorization thing. The first step from PRPP that gets converted into IMP, that first step is going to be your committed step. And it's going to be the one that's heavily regulated and the slowest. Does anyone have questions about that? No. Okay. Now we're moving on to the other direction. So we have our PRPP. Now we're going to go the pyrimidine route, the bottom one, where we're going to form our precursor base first rather than doing the stepwise addition like this, where it adds to the ring itself. So with pyrimidine synthesis, as you can see here, we're going to form the ring outside, and then it's going to ultimately get attached to the PRPP right here. We're going to make this orotate. This is a good intermediate to know. The orotate is simply this molecule right here, and that's your fully formed base. The orotate is a fully formed base. And then from there, it gets attached to your PRPP, the sugar part and the nucleotide itself. And then you're first going to end up forming, I'm pretty sure it's UMP is the first thing right here. And then then from there you can form your cytosine, the C part. Because it undergoes a transamination reaction right here. So you can see the carbonyl group changes into an amine group. But anyways, point is that it first goes from the PRPP, you add the orotate, and it ends up forming your UMP. And then from UMP, it could either go two directions. It can either go to cytosine, or it can go to thymine. And we remember that thymine is that methyl group attached, so we're going to methylate it, and that's for DNA. It'll stay at UMP if we're doing RNA. And I wanted to say that the first step is the heavily committed, no, not this first step, but the formation of carbamyl phosphate is our rate limiting step. And carbamyl phosphate is going to be part of this like cascade. And that carbamyl phosphate formation isn't shown here, but I do want to say that that's the rate limiting, and it's different from the urea cycle. In the urea cycle, we do form carbamyl phosphate, but that's in the mitochondria. In this one, we're using a different isoform. We're using carbamyl phosphate synthetase 2. Synthetase 2 is in the cytosol. The other one is carbamyl phosphate synthetase 1. So they're different isoforms. They're regulated differently. Again, it goes back to that enzyme multiplicity thing. So in this case, it's located in the cytosol. It's going to act differently, and the carbamyl phosphate is now also in the cytosol. It's going to do different things. And when it's in the cytosol, it's going to participate in nucleic acid development, specifically with the pyrimidines. And so that's the heavily regulated one. And CTP is going to inhibit part of the carbamyl phosphate formation, but also the first step. It's incorporation with aspartate. And the reason why is because CTP is a downstream product. We're going to do product inhibition. Does anyone have any questions about this like pathway? It's just more bigger picture stuff rather than trying to memorize every single type of enzyme like dihydroorotate. Like that's not worth your time. It's better to know that you're making the ring. The full ring is the orotate. The orotate's going to get added to the sugar background, the backbone, whatever. You're going to form your UMP first. You phosphorylate it to UTP, then it gets converted to CTP. That's pretty much it. Okay. Another thing also, before I move on, it is good to know where your carbon sources and nitrogen sources are coming from. That is another thing that, again, they could just blank out these terms and then just have you write them in. So that could be a way you get tested. And then now I want to talk about pyrimidine synthesis regulation. So we talked about CTP. I'm not going to really go over that. It's just this first step that's inhibited. I do want to talk about how ATP will inhibit CTP and then how that affects ATCase. So ATCase is this blue rectangle. It just has a different name. They're the same enzyme right here. But I wanted to show you that ATP is going to inhibit the action of CTP. So just let's start from the very beginning. Let's say that there's no ATP. There's just CTP. It's inhibiting ATCase. So this arrow is at 0% function, right? This arrow, it's not working. So we're just stuck with a bunch of substrate now. Now let's say we have a bunch of ATP. The ATP is going to come in and inhibit the CTP. So now ATCase is working at 100% functionality. This arrow right here, this red one that's going on to ATCase is no longer functioning. So now that this is active, now we're converting our substrate into product. And you may say, well, why does ATP do this type of thing? Why does it almost like stimulate CTP formation? And that's because we want to have relatively equal levels of pyrimidines and purines. That's exactly why we have this type of regulation. And I do want to emphasize with this KM curve, with this Michaelis-Menten equations, or graph, sorry. Because they may ask you, like, for example, they'll blank out these terms, which is what they did on my exam, and I was very pissed because I was like, this should not have been tested. But they blanked out these terms, and then they had you basically write the conditions. They gave you the 3 conditions. You just have to ascribe it to the correct curve based on your knowledge of inhibition and activation. So it is good to kind of correlate the two for this case. Any questions on that? Okay. Now moving on to the sugar part, where if we have deoxyribose or ribose, how do we convert between the two? And the story is that you have ribose, which has this OH, and you're going to reduce it and convert it into deoxyribose. So the enzyme name is called ribonucleotide reductase. So usually to form DNA, we have to go through like the RNA version first in order to convert it to DNA. And then the biggest things you really have to know is not the alpha subunit, beta subunit. You just have to know the active site features, and okay, perhaps it's on the beta subunit. That's pretty much it. Is you have the sulfohydryl pair, which is the dithiols, and you have the tyrosine free radical. And you do kind of have to know the mechanism. I have it on a couple of slides. I saw Solomon was like going through it extensively through each step. So unfortunately, like that is like one of the mechanisms that you guys do have to know. But before we get into that, I just wanted to go through the electron flow of this enzyme. So for this enzyme specifically, we have two cofactors for this. We have NADPH, which is going to power the thing, and this makes sense. Like the energy doesn't just come from nowhere. It's going to come from that pentose 5-phosphate pathway, because remember, that's the pathway that forms NADPH. So the NADPH is going to come in here, and it's going to transfer these electrons, and you don't have to know these specific enzymes. You just have to know that it ultimately transfers to glutaredoxin. And that, you can think of it like the iron sulfur crystals, like in ETC, right? The iron sulfur crystals, where you had the NADH go in, and the electrons are transferred to those crystals, and then onto quinone, whatever it was. And in this instance, we have NADPH giving those electrons the energy, and then it gets transferred to the glutaredoxin, which acts as your intermediate, and then it gets transferred to the enzyme, which then becomes reduced. Now that it's reduced, we can turn our ribonucleotide into a deoxyribonucleotide. And that's how this electron flow works. You don't necessarily have to know all these specific things. You just have to know the intermediates. So basically what I highlighted in this magenta box, to start the intermediates and the end result, which is a reduced enzyme. Any questions? Okay. So this is the mechanism itself. I pretty much have everything I'm going to say for the mechanism in words already, but just kind of going over, you start on the top left. You have your tyrosine radical, which is going to add itself, or it's going to donate an electron. It's going to grab the hydrogen, so it's going to do this like almost exchange. Then from there, because that radical creates instability within our ribonucleotide, your thiol groups in the active site of this ribonucleotide reductase is going to, or sorry, the OH on the base is going to attack the sulfohydryl or the thiol group. And what ends up happening is it'll grab the hydrogen, and this becomes a carbocation, like this positively charged oxygen. We know that that's not what it wants. So what ends up happening is the oxygen leaves. And when that occurs, we form a carbocation. But this carbocation is stable enough, and I don't know necessarily the reason why, like the orgo, but because I forgot. But essentially what's going to end up happening is that S group that we deprotonated, it's going to grab onto the other hydrogen from the thiol, or it's going to basically oxidize the last thiol group, and the hydrogen is going to attach itself to the carbocation. And that's exactly how we form our deoxynucleotide. So now the 2' carbon from the sugar no longer has the OH, it only has the H now. So we see it's reduced, and then it just ends up leaving and forming the product. And again, we still have that radical that we formed from the first step. So all we're doing is just getting rid of the radical by grabbing back the proton from the tyrosine. Again, the whole point of that tyrosine is to create instability within our substrate so that this whole reaction can take place. And then it just grabs back the hydrogen. That's the whole function of like the tyrosine in this case. Any questions on this? Okay. And then this is one of the most complicated diagrams that I've ever come across, but I'll try to break it down as best as I can. So don't try to overlap the two. Keep them separate. In the same way that I told you guys for exam 2, don't try to combine gluconeogenesis, or the glycogen synthesis and all that stuff. Don't try to combine these two different distinct pathways. So for the first one, we see here on the left, we have the primary regulatory sites, which is just universal regulation. So remember, This is deoxy. This is the deoxyribonucleotide. So this is the after the fact. And this is regulating the ribonucleotide reductase. So we're forming RNA to DNA. And this is our DNA, this is our RNA. Or sorry, the starting is the RNA. So when we're talking about the primary regulatory sites, right? What we end up happening is the ATP, and this is universal, where it stimulates all of these. And why is that? Because the ATP is a ribonucleotide itself. It's another name for it is adenylate. And adenylate has that 2' OH. And because it's a ribonucleotide, because it's an RNA, I guess you could say, it's going to stimulate ribonucleotide reductase because we want to form DNA. So it's almost like a substrate in a way. So that's why it stimulates. And then when we have DATP, the deoxynucleotide, the deoxy ATP, it's going to do feedback inhibition and inhibit the rest. This makes sense. This is very intuitive. What doesn't make sense is the substrate specificity sites, and that's why I'm saying keep them separate. In this instance, we have regulation at the substrate specificity sites, which just means that like if DTP, and this is just gonna have to be straight up memorization. I'm so sorry, guys. I wish there was a better way to explain it, but it's confusing because some of these don't make sense. Like DTP negatively inhibiting this makes sense. That makes sense. And then it activates DGTP, but we know that T binds with C, not necessarily G. And the whole point of this substrate specificity is that you want your complementary to be promoted and the same one inhibited, if that makes sense. So like, for example, where is it? So like DGTP, right? We see that it does stimulate the formation of DATP, and that makes sense. A binds with G because they're complementary. But there are some instances where it just doesn't, and that just has to come down to memorization, knowing those different exclusion. I'm sure there's an explanation, I just don't know off the top of my head, and I've asked profs, and they aren't able to give me a coherent answer. But do you have anything in terms of that?
No, it's brute force.
Yeah.
Brute force memorization.
Okay, and then I wanted to talk about specifically this, more so the fact of forming DTMP from DUMP. Now, I just wanted to highlight on this top right corner, we have DUMP, and then we have DTMP. And as you can see, the only difference between the two is that you're adding a methylene group, a methyl group here, CH3. And the way you do so is through the thymidylate synthase. And this enzyme, what it's going to do is it's going to do a transfer of a methylene group right here on the gray from our N5, N10 methylene tetrahydrofolate, just THF for short. It's going to transfer from THF to our DUMP, and it's going to form DTMP. That's realistically all you have to know from this. The cofactor is THF, which is just this thing. And the THF has to regenerate via this other like pathway where we have a reductase to reduce it first, and that powers it so that we can regenerate the methylene. That's pretty much all you have to know. You don't really need to know these names, just know like the function, the purpose of it. So again, to reiterate, when we end up converting THF to, I think it's DHF because it's di, when we form our dihydrofolate, we want to regenerate it. So the first step we're going to do is dihydrofolate reductase. You do have to know this enzyme. And what that's going to do is essentially reduce it. And that's going to power it because we know reducing things powers things. And once it's powered up, we can essentially add the methylene group. We can form these carbon-carbon bonds. And that's energetically, like it's an anabolic reaction essentially, and that requires energy. So that's why the first reaction happens in the first place. It's to power the whole thing. And then now we generate our THF, and it undergoes the same reaction. And then two things I wanted to know is that these chemicals they act as chemotherapeutic agents because if you inhibit DTMP formation, you don't inhibit RNA formation, if that makes sense. So I'll explain. In other words, when we inhibit DTMP, we know that's used for DNA. We know uracil is used for RNA. So if we target the thymidylate synthase in some way by inhibiting this reaction, we know that we can inhibit the DNA without inhibiting RNA expression. That's the difference. So the cells can still do their thing, but they won't replicate. And that's what some of these therapeutic agents are aiming to do. So the first one is fluorosil, which you guys should know inhibits the thymidylate synthase, so DUMP won't even bind in the first place. And then you don't need to know mechanism of or whether it's competitive inhibitor or non-competitive. You don't need to know that. And then another one is methoxytrate, which just inhibits the regeneration. So you'll just be stuck with DHF rather than THF with this one. And that's pretty much all I wanted to cover with this because I know it was covered a little bit more in depth in your lecture. Does anyone have questions? Okay. So, what is it? Okay. So in terms of degrading these nucleotides, right? There's two routes. In the similar way how we're making these nucleotides, there's two routes. And when we're degrading them, there are also two alternative options. There's either removing the 5' phosphate group, which we know that if we remove the phosphate from a DTP or an NTP or NMP, whatever you want to call it, it's a nucleoside now. It's just the sugar and the base. Now, if you remove the ribose, you end up forming a free base. So those are the two options. So you can either form a free base or you can form just a nucleoside. And then with purines, they're ultimately degraded into uric acid. And overproduction of uric acid causes gout. And I don't think you really need to know specifically this inhibition enzyme, but if you have time, like at the very, very end, definitely worth something to take a look at. I don't think it's necessarily worth your time though right now. And then moving on to pyrimidines, it degrades into ammonia and carbon dioxide. And this kind of makes intuitive sense. N atoms are ultimately excreted as urea. The C atoms are either converted into carbon dioxide or they're converted into acetyl-CoA, succinyl-CoA to enter the TCA cycle. They're reused in some way. And then another note is that with the pyrimidines, in the same way how we built it not stepwise, but we built the precursor and formed DUMP and then DCMP, we have to go the opposite direction. We have to do DCMP, DUMP, and then break it down. So that's like a thing we have to keep in mind as well. And then I think this is pretty much the last slide. Yeah, is salvage pathways. So there are ways to recover the free base in case we break down too many. And so with recovering purines, we combine them with PRPP. And there is a special enzyme. I know I said that with purines, you build them up stepwise, but we're just combining two of them together. So if we have a free adenine base and a PRPP, we can use a special enzyme called a adenosine, and the main part that you want to memorize is the phosphoribosyl transferase. That, and then obviously it's going to be like whatever the precursor is in the initial name, but it's this enzyme that attaches the two together, and like forces it, and then you form your AMP, IMP, or GMP. That's pretty much all you have to know. I don't think they'll ask you about the middle one here. They'll more so ask you about like the salvage pathway in terms of salvaging adenine or salvaging guanine. And yeah, again, recycling free bases is just much more energetically efficient, but sometimes we break down those bases so they may not even be there in the first place. So then in that case, we build them up stepwise. But that's pretty much it, guys. I don't know if you guys have any questions. I'll stick around. But I'm wishing you guys luck on exam 3. It's much more bigger picture than you think, rather than the smaller details. It is good to know the smaller details, but it is much more bigger picture.